you want to tell them? Yeah. Go.
At this time, Anne Rancourt, Communications Director in the Office of Women's Health, will introduce our next keynote speaker.
Hi, everyone. Welcome back. I am thrilled to introduce our second keynote speaker today, Dr. Londa Schiebinger. Dr. Schiebinger is the John L. Hines Professor of History of Science at Stanford University and directs the EU-US Gendered Innovations in Science, Health and Medicine, Engineering, and Environment project. She is a leading international expert on gender in science and technology, and over the past 30 years, her work has been devoted to teasing apart three analytically distinct but interlocking pieces of the gender and science puzzle: the history of women's participation in science, the structure of scientific institutions, and the gendering of human knowledge. She has twice addressed the United Nations on the topic of gender, science, and technology, and has worked with the European Commission on a number of projects. She's also authored numerous prize-winning books on women and gender, and her research has been featured in the New York Times, New Yorker, on NPR, and in numerous international publications. Her talk today is Gendered Innovations, Analyzing Gender in Preclinical Research. Thank you for being with us.
Thank you. It's a pleasure to be here. And as a professional historian, I think I can say we are making history at this moment. So let's do a good job. Doing research wrong costs lives and money. And you all know this. For example, 10 drugs were recently withdrawn from the U.S. market because of life-threatening health effects, and 8 of those pose greater threats in women. Not only did these drugs cost billions of dollars to develop, but when they fail, they cause human death and suffering. So we can't afford to get it wrong. Doing research right saves lives and money. An analysis of the Women's Health Initiative hormone therapy trial found that for every dollar spent, 140 were returned. And the study also saved lives, 76,000 fewer cases of cardiovascular disease, 126,000 fewer breast cancers, 145 more quality-adjusted years, and the only downside was more osteoporotic fractures. So we know this already. But I want to advocate very strongly that NIH develop metrics for understanding the cost in dollars and in lives to integrating sex as a variable in preclinical research. Now, we started looking at the economics at Stanford. We had a workshop a couple of weeks ago, and a number of people here were involved. And this is just the beginning, but this is what we hashed out after a couple of weeks on email after our meeting. So the question is, will analyzing sex as a variable cost more money? People tend to consider this only in relation to their own lab. And we need to look at it from a U.S. and perhaps even an international point of view. Developing a new drug now costs $5 billion, and the cost is so high because it factors in the failure rate of drug candidates, which is estimated at about 95%, with most failing in phase 2. So the cost of sex inclusion in preclinical research is small by comparison. So back to the question, will analyzing sex as a variable cost more money? And we found, or we are going to do a larger study, but I think NIH should take this on because we aren't going to do it all. So there are 2 things you have to look at. The effect on sample size, and we discussed this a bit this morning. Adopting a strategy of 50-50 female and male animals or cells instead of 100% of one sex seems likely to allow detection of at least some sex influences, namely the largest ones that presumably researchers would first want to detect, with not much impact on sample size. So in some cases, additional sample size may be valuable to study sex differences in finer detail, studying variants such as age or reproductive phase, for example, in males and females at the same power. And even in this latter case, sample size may not need to be doubled. So the second effect is on study cost. Many costs of running studies are fixed, such as basic lab operations, and would not vary with the sex of included animals or cells. And some costs may in fact be expected to decline, for example, because housing female animals may be cheaper than housing male animals. An additional statistical analysis and reporting may have some cost, but this is likely to be minimal. So in general, some adding considerations of sex difference would often have small effects on overall costs, though in some situations costs might increase and in others they might decrease. And even when costs increase, they should be weighed against the additional cost. Well, so any additional cost should be weighed against the possibility of new insights, improved therapies, and reduced risk of subsequent problems. And recognizing this issue of costs, the NIH in 2014 distributed $10.1 million to support sex inclusion research. And it's anticipated, I hope, Janine, we can say, that additional support will be provided going forward. So we may also need new legislation. Being a good historian, I went back to the 1993 NIH Health Revitalization Act, Public Law 103-43, Subtitle B, and was shocked that it applied only to women and minorities in phase 3 clinical trials. I somehow had it in my brain that it applied to females in research, biomedical research. So maybe we need a new law that states this latter case. So as we can see, it's crucially important to get the research right, and that's the goal of this Gendered Innovation Project that I directed. It develops state-of-the-art methods for sex and gender analysis, and it provides case studies or examples of where, if you actually use sex and gender analysis, you get something new. So we took examples of things that had already been developed to give people 23 examples of how that is true in a globally accessible website so that you can see it. So let me go there just for a second and show you. This is a good resource for students or for anyone who has not thought about sex and gender. Since it's not included in the curriculum, we need a starting point for people. So over a period of 4 years, this project was funded by the European Commission, the National Science Foundation, and also Stanford, and we worked with 60 basic scientists, engineers, technical people. Virginia Miller was part of our team for, where did we meet? Berlin, or I forget exactly where we met. We met in all these wonderful places. And we hammered out methods or questions that people can begin to put to their projects if they don't know where to start. And some of these are important. For example, rethinking priorities and outcomes. We have to think what our priorities are. Why are we putting public funds in researching a particular thing? Another of interest for us here today is analyzing sex, analyzing gender, analyzing how those two interact, and I'm going to say more about that, and then analyzing all the other factors that intersect with sex and gender. For humans, these include, of course, socioeconomic status, lifestyle. In cells and animals, these include comorbidities, cell types, the type of disease being treated, animal strain, and caging practices, and so forth. And then you can also see our case studies in these different buckets. Someone was mentioning today this osteoporosis is an instance where men have been left out of the research, and so we delved into that one. So, and I'd like to reinforce here just one point from our method analyzing sex, and this was also touched upon this morning, and that is reporting null findings. This is very important, and I think journal editors need to hold the line on this. So Marcia McNutt, who is the editor-in-chief of Science, tells me that in the first week of November, we will have these new guidelines for researchers. I hope they're really, really good. And let me just give you a quick example from Stanford. Our Center for Sex Difference Research has started giving seed grants to researchers. So whatever they're doing in their research, if they add the sex or gender dimension, we will give them some extra funding to do that. And so the first round of funding is coming in now. And one of the labs was panicking because they said, oh, we don't have a result to give you. And the PI simply didn't recognize the absence of a difference as the result. And so we have much more education to do. So people have to recognize that, and journals need to publish it. Just a second. So I want to go back here for a second. So now I want to open the discussion to gender. Sex refers to biological characteristics, and gender refers to sociocultural processes. Authors in peer-reviewed journals use the word gender because they don't know what it means when they mean sex. So my point here is that the words sex and gender are not interchangeable. I'm preaching to the choir here, but we have to get this on to our colleagues. Each is a technical term and needs to be used precisely. Peer-reviewed journals must hold the line on correct terminology. Careful use of these terms will enable meta-analysis of published and archived data, which at the moment is not really possible. So we as a research community really are smarter than that. It's smart. We shouldn't be confusing these things because it's not that hard and we can get it right. Okay, now on to the gender in animal research. So gender is a constellation of sociocultural processes that interact with and thus influence biology. So that's why we care. The danger is that PIs confound sex with gender, and I don't mean here just using the terms incorrectly, but that we see sex or a biological trait when in fact we are looking at gender in the lab or at environmental conditions. So gender influences sex and vice versa. In animal research, I will use the term gender for the subset of care, management, and environmental processes that may impact male and female animals differently, such as caging or differential handling of male versus female animals. And gender then will be those characteristics that could be mistaken for sex. So it's a subset of the environmental factors. I don't want to just call it the environmental factors. So again, we don't want people to see sex or a biological trait when they're really looking at gender or an environmental condition. And the reason I call this gender and not just environmental factors is that these gender differences are precisely the ones that we might mistake for sex differences. So that's the challenge. And I would argue that analyzing gender in addition to analyzing sex will be important to increasing the translational value of animal models. So let's have a look. So I've just had my visualization guy at Stanford start working with me on this, and I'd be happy to get your responses. So animal research includes what is in the animal here, analyzing sex, those are the biological traits, and then how these interact with what I'm going to call gender. I have an example for each of these, but I'll only talk about two. And the double-ended arrows show the interaction between the two. So in general, the term gender has been applied to humans. However, it might influence animal research as follows. And I want to make three distinctions here. So gender norms, and you have to learn this. I'm sorry, I'm giving a quiz afterwards, so you have to learn this. Okay, so gender norms refer to researchers' attitudes toward and handling of male and female animals. Researchers may act on gender stereotypes concerning the behavior of male and female animals. So gender norms are the forces. For humans, they're the social forces acting on you to make you follow the rules. And I think for animal research, they're the forces, the gender norms are the forces from the researchers' attitudes and behaviors that are acting upon the animal that might trigger stress or particular behaviors in the animal. So we'll look at an example of that in a moment. Gender relations refers specifically to the interactions between male and female animals on the one hand, and between men researchers and women researchers and male and female animals, so between researchers and animals on the other hand. Now there's also gender identities, and this is what you're most familiar with. This is what your undergraduates will think of when they're thinking of gender. And gender identity refers to how individuals perceive and present themselves and how they're perceived and presented by others, whether their behavior is considered masculine or feminine, straight, trans, or cis, for example. Facebook last year released 51 categories of gender, so we can't just be binary now. So I'm saying that I don't believe the gender identities applies in the animal lab. I do not believe that little mice and rats have gender identities. I think that Virginia's primates might. I think that some of the other animals might, but not these animals. So I'm interested in the first two, the gender norms and the gender relations. Here's another, I think visuals are very helpful. Here's an image of how sex and gender interact over the course of the lifetime. So you have, even in the germ cell, you have both coming together, and then you have the biology happening, interacting at the same time at every stage of the life to influence the adult animal or the animal as long as it may live. So let's look at a couple of examples of how gender might be confounded with sex. And some of these have come out of, I think, interestingly, come out of Canada, because Canada, since 2010, has had a policy for integrating sex and gender into their basic research. It doesn't have teeth because you can still get the funding even if you don't do it, but I think it has sparked a lot of research. So the first example, and let me just go back to the blue mouse, I want to talk about caging, an environmental effect, and the animal. So importantly, differences in caging practices between male and female animals should not be mistaken for biological sex differences. Now, we know that males are often caged alone because they fight, and females are typically caged 3 to 4 in a group because that saves money. Ritz et al. Out of Canada shows that when animals are housed alone, they expend more energy maintaining body temperature, which can cause differences in parameters such as caloric intake, muscle activity, metabolic rate, fat distribution, or body size, and then a plethora of potential downstream effects on bodily and cellular activity. Animals housed together, by contrast, tend to cluster together to sleep, and as a result, they spend less energy just to stay warm. So the danger is that if we don't analyze gender, sex difference may be identified where, in fact, differences result from housing conditions. And it's important here not to see a sex difference when what we're looking at is influenced by caging difference. Now, we all know this, but Prendergast et al. In their 2014 meta-analysis found that half of the studies of mice failed to specify the number of animals per cage. So there's a lot to be said about caging, but you get the point. So I want to go on now to the research staff with a second example. This example was mentioned already this morning, but I want to go into it in more depth. This is an example about the sex of the researcher and the response of the animal. So it's not about the gender of the researcher. It's not about the attitudes or behavior of the researcher, but it's about their pheromones. And the gender in this example is then the relationship between the researcher and the animal's response. So it's in that interaction that I was talking about before. So here's the study. Experimenter sex may be a confounding variable in rodent research where stress is a significant factor. Sorg et al., and this came out of Jeff Mogel's lab at McGill earlier this year, found that rats and mice demonstrated a reduced pain response in the presence of a male experimenter as compared with an empty room, whereas the presence of a female experimenter produced no differential response. Both male and female rodents showed this response, but the effect was actually greater in females. Researchers identified what they called male observer effect as a stress response to male pheromones found in higher concentrations in men than in women. Stress, as we know, can affect the release of gonadal steroids and sex differences in behavior and physiology. So in this experiment, researchers were specifically measuring pain response in the animal. In this case, they found that the sex of the researcher matters, but it's an example of gender relations that needs to be taken into account. Animals had specific responses to men and women researchers. And according to Mogul, this may throw into question all prior results from pain research. So let me conclude with just a few comments about policy, because I'm very interested in the policy that we are developing here. And on our website, we also give a lot of information about policy. We have policies of major granting agencies and peer-reviewed journals. So I want to do 3 interrelated areas. So granting agencies are very important. Granting agencies can ask applicants to explain sex and gender analysis and how this is relevant in their proposed research as a requirement of funding. And I have to say the U.S. is a bit behind in this. We were ahead in 1993 when NIH asked for women to be included in phase 3 clinical trials. But now, I think the European Commission has moved ahead. In December 2013, they launched Horizon 2020 and designated 137 subfields of science, health and medicine and engineering that must include gender and sex analysis. And as you know, Horizon 2020 is the next 7-year funding framework for the European Union. And as I already mentioned, since 2010, Canada's Institutes of Health Research request all granting applicants include sex and gender in their research design. The EU, the EC, has more teeth than Canada, but I think Canada has been important here. So I think these policies for NIH are very important. We need to keep our place in the world and we need to do research right. Among other things, I might recommend that NIH develop some sort of compendium of sex differences. I draw your attention to the Berlin Institute of Gender in Medicine. On their website, they have a database of vetted articles on sex difference in humans and in animals. And I think that we should take this opportunity to sync up with international groups that are already doing some of this work. The Karolinska Institute in Stockholm also has a lot of resources. So that's major granting agencies, and on this website, or on this page, we've given the policies of the granting agencies that we know about and that we think are important. Now I'm going to mention peer-reviewed journals, and on this page we give all the policies of journals that we know of. I'd be happy to add more journals to this. So the second area of policy important to implementing sex and gender analysis is the peer-reviewed journals. Editorial boards of peer-reviewed journals can require sophisticated sex and gender analysis when selecting papers for publication. And I think the very best policy right now is from the Clinical Orthopedic and Related Research, this journal. In January of this year, they did a wonderful editorial. If your journal is thinking about this, I think you can adopt it to your particular area of research. It's really useful. And then finally, we need to integrate the knowledge of sex and gender differences into the medical curriculum. I think it might be criminal if we don't. Aren't people going to die if medical students don't have the right information? And I know Virginia Miller has been working on this. We at Stanford would like to join in. In Europe, there is a consortium of 7 medical schools that are working on curriculum, and I think that maybe that we certainly need to do more. So thank you very much.
We'll entertain any questions.
I just would like to point out that I'm pretty sure Endocrinology, which you can't quite see there, when Jeff Blaustein was the editor, actually adopted the rule that the title must state if it's male or female. He is leading the charge. The bigger journals that I've interacted with don't want to go there, but I certainly hope they understand that they need to go there sooner or later.
I just want to make sure that credit is given where credit is due. I mean, the major driving force for the change in curriculum has come from the Women's Health and Gender Collaborative, spearheaded by Marjorie Jenkins. and her materials, which are going to go live pretty soon.
Oh, good.
She's going to put out an RFA for medical schools to actually beta test their materials. So she's going to offer grant money for them to incorporate those materials into the curriculum in the spring, to have an analysis of that success for a workshop we're organizing jointly with the AAMC, and there'll be more information coming out on that. As soon as the licensing programs start asking the right questions, we think that that'll be facilitated. But in the meantime, I think the comprehensive material that she has developed, it's going to go across specialties, including pharmacists and nursing and health science, as well as medical schools, is really going to be transformative. All the materials have been vetted through experts, and it's going to be, Larry introduced it's going to be a great web material and definitely complement the Gendered Innovations, which has led the way in terms of providing international resources.
Well, that's really good to know. I can't move Stanford on this point. I just can't move them.
I just wanted to add to that besides trying to get that into the medical curriculum, there has to be a move to get that into the graduate curriculum as well.
Absolutely.
We already have the requirement for the responsible conduct of research that every grad student on NIH grants has to take. I think this should be a part of it.
Mm-hmm. Yes, we have to train the next generation. This is absolutely crucial.
This is kind of a curiosity question, but maybe it'll provide some insight. Why is it that both Canada and Europe have been so much farther out front on this issue than we have?
Well,
we're the United States of America, after all.
Well, and we invented gender analysis. North America did. I think Canada and the U.S. But we don't have any stomach for policy, and everyone says we don't want to do things top down, and I think that's really where Europe gets its strength. So I should say that Europe tried to do this in 2003. They tried to require that people integrate sex and gender into research design, and it failed. They got too much pushback because people didn't know how to do it. So as soon as they kind of learned what I was doing, they arrived in my office and offered me money. How good is that, right? But they wanted to develop tools for this. So now I've told them the next step is to have big training workshops or something. I think NIH, I don't know if that's a model for you, but big training workshops to get the current generation of researchers up to speed. The things, I mean, it's not rocket science, but we have to do it right, and I just can't believe that such smart people don't get it right.
Londa, congratulations on this talk and your work. And can you update us on what's happening at the UN? Maybe update us on the testimonies you did and maybe what's happening, what's on the horizon there.
Yeah, so I don't think so. The UN is not really the agency to take this on. They have so many large issues. But the year that I worked with them, they took gender in science and technology as a main theme for the women's unit. And we did a position paper. I had to do all of these issues globally, which is really, really hard. And then just last year, they had an update, what had happened. And I was very happy to be able to report on the European Commission. A lot had happened there, right? So I think we have a lot more to do in the U.S. I think, so now NIH is on board. NSF needs to get on board. I have great hopes with France Cordova, who used to be my colleague at Penn State. And she gets it completely. And I don't know how but so all of the life sciences at NSF, it's very important. And also for any engineering that has a human endpoint, these issues are extremely important. I want to tell
I'm a
Google
program
that.
director in NIGMS, and I just wanted to say that we recently put out a funding announcement for institutions to come up with training modules for incorporating sex differences as a biological variable.
Oh. Yeah. So what can you just say a little bit more about what this is?
Well, it depends on what the institution comes up with. So they have to put in an application,
right.
And then it
Great.
gets reviewed. So we put out a feedback loop on this. I don't know, maybe about a month and a half ago.
Okay. Maybe I could get your card out.
Send it to Janine, and maybe she could distribute it.
I would like to get, or I could get your card or something. I'd like to get the details.
Thank you. We'll move on to session 3.
I'm going to pull it up.
Okay, we'd like to invite all the speakers for session 3 to come forward, please.
He changed his talk. So, did you get the right one for me? Which one is it? This one.
Officer Research OW.
I got the one.
C on my thumb drive.
Okay. Is this alphabetical order? No.
Is that my thing?
Mm-hmm.
If you click on, it's the most recent file under date. If you click on date.
Okay.
You click on the top here, or maybe click on date, and then click it on it again. Click on it again. That's it.
Okay.
But on this next second.
Yeah. All right.
I made some changes to my slides.
Did
so
you?
If I can just flip in here, that would be great.
Yeah, okay.
Computer, I have my dongle.
You have it? Oh. That I'm not sure of.
Doing it now?
Yeah. Okay. How does that work?
The computer.
I don't know how to hook this up. Let me grab the guy.
I'm gonna grab him. Okay. Where am I supposed to hook this up?
Do you know?
Okay, so welcome to session 3. We've had a lot of interesting presentations today that really point out the importance of conducting a sex-gender analysis of data. And while the focus has been on preclinical research, we've heard it's also important in clinical research as well. So we're going to be moving into session 3, which is entitled Practical Methods to Integrate the Biological Variable Sex into Research Projects. So if you had an earlier version of the agenda, you may have been expecting Gary Churchill here. I am not Gary Churchill. I will be the moderator. I'm Coralie Wetherington from the National Institute on Drug Abuse. And this general topic has been one that NIDA has been advocating for a number of years. We had a program announcement in this area and have continued since 1992, but it's a really hard nut to crack, as many of you know. But the Clayton Collins article, I think, is just a major turning point for us. So we've been advocating for looking at sex differences in preclinical research and in our clinical research in all areas of drug abuse, and to analyze by sex gender in all areas, and also to recognize that when a difference is found, you don't just leave it there as an interesting observation, but you do something about it. You take it to the next level, which may be an application level, or if you're talking about basic preclinical research. It may be what's the mechanism, what's going on here. Whenever you see a male-female difference, you've got a free tool there. It's telling you that if you can find out what's causing the difference between males and females, you're learning something about the subject matter that you're studying. And we also recognize that when there is no sex difference, no gender difference, that doesn't mean that sex or gender does not matter in that particular subject matter. And so males and females can get to the same endpoint, but by different mechanisms. And that's just a really important point to drive home, that finding no difference doesn't mean that being male or being female does not matter. And another point is that just simply not finding a male-female difference, say in preclinical research, if you throw in another variable, that may change the whole thing. That may reveal the male-female differences. So these are the types of issues that we have been advocating for researchers to recognize for a number of years. So in this session, we have three speakers, and I'm going to take some time now to tell you a little bit about all three of them and their talk, and then they will speak one after another. So the first speaker is Dr. Susan Makris, who's a toxicologist from the Environmental Protection Agency, where she's been working in various capacities for the past 23 years. Her regulatory work has focused on toxicology testing and risk assessment guidelines, guidance, and policy as applied to the assessment of developmental and reproductive toxicology and risk. Initially at EPA, she was in the Office of Pesticide Programs, where she evaluated hazard and risk characterization for numerous pesticides. Since 2004, she has been with EPA's National Center for Environmental Assessment, where she is a senior staff toxicologist in the area of human health risk assessment. She's authored numerous journal publications and book chapters in this field and is a long-standing member of the U.S. and European Teratology Societies, the Neurobehavioral Teratology Society, the Society of Toxicology, and the Society for Risk Assessment. In her talk today, Dr. Makris will discuss the toxicology guidelines that the EPA uses in preclinical research, with particular emphasis on methods and techniques for integrating the biological variable sex in that research. And then she will conclude with a discussion of the importance of this sex-based approach. The second speaker will be Dr. Art Arnold, who is a distinguished professor of integrative biology and physiology and director of the Laboratory of Neuroendocrinology at UCLA. He is also editor-in-chief of the Biology of Sex Differences, which is the official journal of the Organization of the Study of Sex Differences, which is a publication of the Society of Women's Health Research. He's also the inaugural president of the Society of Behavioral Neuroendocrinology. That was from 1997 to 1999, and he's a AAAS fellow. He has long studied biological factors that make males and females different and is interested in what he calls the sexome, which he describes as the sum of all sex-based influences on gene networks in cell systems studied globally. His talk today will highlight the fact that for two prevalent and serious health conditions, obesity and heart disease, research has not asked the fundamental question of whether our sex chromosomes play a role apart from our gonadal hormones. He'll describe his four-core genotype model as a way to tease apart those two factors. And as a spoiler alert to women, you may not be happy with all of his results, but remember a comment made earlier by Jill Becker that biology is not destiny. And then our final speaker is Dr. Jillian Einstein, who's a faculty member in the Departments of Psychology and Public Health Sciences at the University of Toronto, where she is director of the new collaborative program in women's health. She's currently treasurer of the Organization for the Study of Sex Differences and serves on the advisory board of the Institute of Gender and Health, which is one of the Canadian Institutes of Health Research. Her book, Sex and the Brain, A Reader, published by MIT Press, is a compilation of classic papers in the field of hormones and behavior with critical introductions to each chapter. Dr. Einstein's research program focuses on women's health research, sex differences in the brain, and the empirical exploration of whole-body explorations of mind. Her work is based on the premise of brain plasticity, and she uses a variety of methodologies, including standard psychological tests of memory and pain response, psychophysiological tests, genetic tests, steroid hormone assays, fMRI, and qualitative interviewing. Her talk today will center on 3 topics. First, what clinical researchers need in order for animal experiments to be relevant. Secondly, we'll hear more about integrating the idea of gender in preclinical studies. And finally, she will tell us about some approaches from Canada. So, our first speaker.
Thank you for the very nice introduction, and I would just like to thank the organizers as well for inviting me to this really wonderful workshop. I'm just really impressed with everything I've heard today, and with all of the initiatives that are currently ongoing. They're just really astounding. A lot of work, a lot of really thoughtful conclusions that people are coming to. So. I had, as you were told, the task of talking about toxicology testing guidelines, actually GLP preclinical testing guidelines. And in a way, it's a little bit, maybe a little bit off track of what some of you guys are doing in your life and in your jobs, but there is a lot of overlap as well. So hopefully I can bring that to your attention today. There we go. Okay, so testing guidelines are standardized testing protocols. I don't know if you've really had a lot of time to take a look at testing guidelines. Probably not. Most of you seem to be researchers, and you're making your own protocols. But these are standardized testing protocols. And they're developed for regulatory agencies through peer review and public comment processes, and they're published. They're publicly available on regulatory websites. And it's not just EPA, it's not just FDA. There's a lot of different agencies that have testing guidelines, but I'm just going to focus today on EPA and FDA. FDA also harmonizes with the International Committee on Harmonization, and EPA harmonizes with the Organization for Economic Cooperation and Development, which is a global group that's based in Paris. So these guidelines really are very broadly applied across a number of different agencies and offices within the agencies. And there are a lot of different guidelines, different categories of guidelines that are listed here, both for EPA and FDA, but a couple of them I've highlighted because they are the ones that are focusing, for the most part, on the toxicology studies, the preclinical toxicology studies that we're interested in today. So for EPA, they're the health effects test guidelines, and for FDA, they're the safety guidelines. And I mentioned GLP, and that was one of the things I was asked to talk about a little bit. Good laboratory practice regulations are intended to assure, to as much as they can, the quality and integrity of data for regulatory decision making. And so these guidelines include specifications on lots of different aspects of study conduct, on how the studies are organized, the actual organization of the laboratory and the personnel, the facilities, equipment, and animal care, and standard operating procedures that the laboratory has to have on hand, how the test and control and reference substances are handled, the study director and what their roles and responsibilities are. GLPs require that every study be conducted with a quality assurance unit that's based in that laboratory that looks at both the conduct of the study, the protocol for the study, and the reports that are done for the study as well. GLPs cover documenting the data, recording it, archiving it, and reporting it. So even the reports are reviewed by the QA unit. And that can give you a lot of confidence in the way that the studies are conducted and also the reports that you get from a GLP study. But they actually don't address the adequacy of the study design or how appropriate the endpoints are that are being collected. So it serves one really very important function. You end up with a really good study when it's done under GLPs generally. And but it really doesn't say, well, have we looked at the right endpoints? So another thing that needs to be talked about in terms of guidelines, the one thing that isn't addressed by the guidelines is when would you actually do the study? And for different regulatory agencies, that can be based upon different attributes, different guidelines or guidance or decision making. Sometimes there are toxicology testing requirements, for example, for the Office of Pesticides and the law under FIFRA. Data call-ins might be applicable to some agencies where the agency would say we need this particular study, and a registrant or industrial group would need to provide that data. Test rules can be set up through negotiated processes and very, very formal processes. There can be published recommendations or guidelines. There can be negotiated agreements that happen between industry and regulatory agencies, and sometimes the studies that are conducted might be actually non-guideline studies, although they would very likely be conducted in a GLP laboratory. And this is just an example of the tox testing requirements for food use pesticides, where there are a number of different studies that are required: acute, subchronic, chronic, developmental toxin repro, mutagenicity, and sometimes special testing. And some of the studies listed there are conditionally required, where it would be based upon the data that you have on the chemical. Okay, so there might be different strategies, too, that are being used in the toxicology testing for these preclinical studies. And there are some real differences between the way that environmental agents are approached and the way that pharmaceuticals would be approached. So for the environmental agents, these are actually broad screening studies. And the purpose of them is to identify hazard and dose response for use and ultimately in risk assessment. And a lot of times going into these studies, there's really a lack of some really important information, for example, mechanistic information, mode of action, or adverse outcome pathways. A lot of times toxicokinetic data are not even available. So that would lead you to an approach where you'd need to more rigidly adhere to the guidelines, to the study protocols that are out there. Also, there's not a lot known sometimes on human exposure. Sometimes the pesticides or the chemical substances that are being tested, we don't really have a lot of information about human exposure, and it might be assumptions that we're using to try and estimate how people might be exposed. We might know what the use is for a pesticide. We might know maybe what's showing up a little bit in the environment, and we'll make some assumptions. But a lot of times human biomonitoring data aren't really available. So, and in general, the purpose of the risk assessments that we're doing for environmental chemicals is to avoid or limit human exposure. So for pharmaceuticals, it's a little bit different because the studies are generally designed to focus on specific questions regarding target organ toxicity and assess the safety. And there's usually a very extensive database of information that's available for an incoming pharmaceutical that's being assessed. All of the good stuff that's missing for environmental chemicals, mode of action information, perhaps, certainly pharmacokinetic data. And so that leads to an approach where there's a lot more flexibility in study design. And of course, for pharmaceuticals, the human exposure is going to be intentional, not accidental. So the tox testing guidelines are designed to evaluate a range of issues. And I'm not talking about just within one study, although some of these issues are within a study, but also across studies in general, the whole scheme of testing. So different life stages are looked at, different durations of exposure and routes of exposure. There are multiple treatment levels looking for a dose response in these studies. We're looking at species differences. A number of different species are used in the testing. Gender differences and multiple target organs, structural and functional effects, and by that I mean the difference between doing pathology and doing some functional aspect of reproduction or immunotox, and also mechanistic data and kinetic data. Okay, so these are the regulatory testing guidelines that are listed for FDA and ICH under the safety guidelines. And I'm not going to go through them in any manner, but one thing I did want to point out here is that within this list of various testing guidelines, there are some of them that are actually guidance documents where they're not specifying how do you do this study so much as they're saying, when do you do this study? What would be the reason you'd want to do a study like this? And some basic principles for doing the testing. And in that way, this is a very different kind of an approach than what's used for environmental chemicals. And it allows a lot more flexibility in the application of these guidelines to testing. Whereas for the regulatory testing guidelines for EPA, the health effects guidelines, there's, as you see, a whole list of various studies by title and each guideline will have detailed recommendations that go into all aspects of study design, every endpoint that's going to be looked at. And I've included here a couple of endocrine disruptor guidelines at the end as well. These are not a part of the health effects guidelines. They're in a different group, the endocrine studies. But I included them here because they sort of illustrate a point in just a bit. And that's here where I pulled out all of for both the FDA and the EPA guidelines, I pulled out the repeated dose mammalian toxicity studies. So this doesn't include the mutagenicity studies, which are more mechanistic orientation, and doesn't include any of the in vitro studies that are in some of the endocrine assays for EPA. And what I looked at was, do these guidelines actually say treat and evaluate males and females? And as you'll see here, most of the guidelines do specify evaluation of both sexes. A few of them don't. A few of them say you could use either sex. Some of them, there's a blank because they don't actually mention that sex, and there's reasons in some cases. I just also wanted to mention, I mean, these are not the only guidelines that are out there. FDA also has Red Book guidelines for safety assessment of food ingredients, and they're actually a lot more like the EPA guidelines. And on their website, they include a general guideline for toxicity studies, which does say that both males and females should be tested in guideline studies. So there's this general approach in these studies to looking at both males and females because it's sort of upfront screening for assessments of environmental chemicals and pharmaceuticals as well. There might be some reasons that only one sex is used in some of these studies. Sometimes it's actually a strategy to reduce the number of animals used. And if we go back again, the studies that, let's see if I can point it out here. I have no control over this. Here, there's some neurotox studies here, scheduled controlled operant behavior, peripheral nerve function, and neurophysiology, sensory evoked potential studies, and those studies are studies where there are a lot of work, there are a lot of effort. They're not used for every chemical, and so they actually say to reduce the number of animals, you could just go ahead and do one sex, animals and the amount of work that's being done. So, but that's really sort of the exception. Often it's because one sex has been shown to be more responsive to treatment because of the kinetics, physiological differences, target organ specificity, or life stage issues. And those kinds of decisions are made for the most part on a case-by-case basis. Sometimes the intended test substance administration is to the fetus, and so it would be via the maternal animal, and the fetus is really the subject that you would want to look at rather than the maternal animal. The maternal animal is the vehicle, and then sometimes, especially for pharmaceuticals, it may be that human exposure is only anticipated in one sex. And this is an example of a study where only females would be dosed on a prenatal developmental tox study, which is the embryo fetal developmental tox study under the ICH guideline. And there, the maternal animal, either rat or rabbit, is dosed during pregnancy, and then a C-section is done. And the evaluation of the fetuses is really the important endpoint of this study, an external, visceral, and skeletal evaluation of fetuses. So this would be an example where the males are not really on study. The males are mated to the females, and then you go forward from there. And we do see some real impact of sex on reference value derivation. And I looked at the IRIS database, which is the Integrated Risk Information System that's maintained by EPA. And this is a database of over 550 chemical assessments, and it's looking at hazard identification and dose response information that can be used in risk assessments, including reference values, and the reference values are generally for lifetime exposures. And I did a search of all the critical effects that reference values were based on in this database, and there were a number of chemicals that came up where there were sex-specific sensitivities that were identified, where the critical effect and the reference value was based only on males or only on females, either for oral or inhalation studies. So inclusion of both sexes in toxicity testing can provide very useful information, and it can identify gender, or shall I say sex-related susceptibility, establish safe levels for pharmaceutical administration or hazard identification and dose response for risk assessment. And then taking that forward, the data from guideline studies can then provide insight for designing clinical or epidemiology protocols, and also for identifying additional research needs. And so I think that's it. Okay. So I hope that that gives you at least some perspective of what we're doing with guideline studies and EPA and FDA for pharmaceuticals, and the impact that that sex can have, and that we're really looking at both sexes very standardly. So if you have any questions. We'll hold. Okay, great. Thank you.
So thanks, Coralie, for that very nice introduction, and I'm delighted to be here. I wanted to say that from the researchers who are working out across the country on this issue, Janine Clayton and her office, and Coralie and others at NIH are real heroes, and we really appreciate the work that you're doing to turn the tide here and to turn the corner and bring sex differences research, increase its salience at NIH. So I have a couple of tasks today. One is that I want to say a couple things about this idea of the sexome, and I also want to talk about sex chromosome effects on sex differences in disease. So I was talking to a researcher at a conference recently, and I had just met her, and I wanted to kind of figure out what she was doing in her lab, and I asked her, what is the main focus of what you're doing? She said, well, our main focus is that we're interested in sex differences in the cardiomyocyte. And then she paused and said, but I never say that in the title of our grant proposals, okay? And the reason was that calling it sex differences research could make it sound kind of less interesting. And she was worried about reviewers who would think that if the focus is on sex difference, it's not as cutting edge as all the other, what Larry was talking about, fundamental mechanisms, which are the mechanisms that males and females share. So I just wanted to say that there is a place, if you are studying sex differences, where you can confess your interest and come out of the closet and publish your research on sex differences. And that's the Journal of the Biology of Sex Differences. I'm privileged to be the editor. It's a publication of the Society for Women's Health Research and the official journal of the Organization for the Study of Sex Difference. So please submit your articles. Okay, the word sexome, this word occurred to me a few years ago, and so I thought, well, for sure somebody had already thought of the sexome, right? So I Googled it, and in fact, nobody, there were no websites that used the word. So I figured if Google hadn't found it, it must be a new word. So we have the genome, we have the transcriptome, we have the methylome, we have the epigenome. We can think of this exome as the aggregate of all sex-biased influences on the biology of the cell and organs and individuals, okay? And it really comes from kind of a systems biology point of view of looking at the system as a system, kind of viewing it from 30,000 feet and looking at the emergent properties of many interacting components examined in using sophisticated mathematical algorithms. So if we think of these green balls as being individual genes that are hooked together and they're pushing and pulling each other, they're increasing and decreasing. You think of this kind of syncytium inside a cell, of this pulsating network, very complicated network of all these tens of thousands of genes talking to each other, that coming in from, maybe not quite from the outside, but you have these female-specific factors. So what is it about being female that's different from being male? And the male-specific factors, and it kind of reaches into the syncytium and pushes and pulls a little bit and tugs on this scintillating mass to alter the emergent properties of cells and tissues, okay? And in our lab, we enumerate what it is about being females that are different and enumerate the sex-specific factors coming from males. So in females, it's having an ovary and ovarian hormones. It's having 2 X chromosomes, not 1 X chromosome. And that makes females different from males. In males, it's having a Y chromosome and only having 1 X chromosome, and having testosterone and testes, and that makes the male different. And these reach into the cells and alter how they work. So this kind of sexome systems biology of sex differences has not actually been applied very often by sophisticated systems biology labs. It's actually something that I would have aspired to, but I'm too busy writing grant proposals. So one lab that has started to do this is my colleague at UCLA, Jake Lucis, and here's just a couple of figures for flavor of how this can work out. His student Tila Van Nass, in her PhD thesis, measured the gene modules that emerge from measuring how genes correlate with each other across individuals. So genes sort themselves out into modules of genes that are co-expressed. They go up and down across individuals together. And this is one of the kinds of correlograms that you can get. And the kind of square things are different modules of genes that are correlated with each other across individuals. And you can see that in some cases, and this is in fat cells, I think, adipose network, that the modules that emerge from just the behavior of genes are different in males and females. And if you use a standard kind of graphing technique to plot the network and to show each of these little ballies is a specific gene, and it's connected by the lines to some other genes with which it is correlated, you can see just kind of get a flavor of it more than anything else. There's nothing quantitative about this, that the patterns of gene co-expression are different in males and females. So we'd like to see more research on this using these kinds of methods and this kind of attitude. And expect that this will be emerging from a lot of different labs. There are just a few papers, really, that do this so far, but there's going to be more. All right, so I want to turn to work from our own lab with numerous collaborators, which is to talk about the components of being, what's different about males and females, and how do they influence physiology and disease. And basically, we're acting as sexual differentiationists here. That is, we're thinking about how can our general idea of what makes males and females different provides a conceptual framework, provides a theory that we can draw on over and over, okay, to find the sex-specific causes of sex bias and disease. And we always start with this basic difference, which is that in the zygote, in the fertilized egg cell, the only sex difference we know of is in the sex chromosomes, having two X's versus one X and one Y. And all sex differences have to be downstream from this. Now, the old theory was that really the only major difference was having a Y chromosome or not and having SRY, because if you had SRY, if you're a male, you have testes and you have testicular hormones, and that makes the male different from the female. The female doesn't have that one gene, so she has ovaries and she has ovarian hormones, and that makes her different. But that model's now been superseded. We now know it's more than just SRY. There's at least 4 categories of different genetic differences that we can think of between an XX and an XY cell, and we can categorize them. I'm not going to go into the various categories because of lack of time, but it comes down then to a more comprehensive theory of sexual differentiation in which is there a pointer? Thank you. Where the fundamental difference, this genetic difference, plays out in gonadal tissues to set up lifelong differences in hormonal actions that account for most sex differences. But at the same time, this genetic imbalance happens in all cells of the body to cause an unequal expression of X and Y genes, which cause these sex chromosome effects, which we've been trying to study. The main problem is, how do you dissociate the hormonal and the sex chromosome effects? We'd really like to take a female and give her a Y chromosome and see if that makes any difference, but as soon as you give her a Y chromosome, you give her testes, and all of a sudden you gave her the hormones too, whereas all you wanted to do was give her, let's say, the genes on the Y chromosome. So the first, it's really 2 major mouse models, and now a whole bunch of other mice too that are downstream from that, but it's the 4 core genotypes and the XY star models. The 4 core genotypes model was pioneered by Paul Burgoyne and Robin Lovell-Badge, and this makes gonadal determination independent of sex chromosome complement. In other words, giving a female a Y chromosome without making her male, in a sense, okay? So in this model, the Y chromosome, which normally has the testis-determining gene SRY, is deleted for SRY, and the SRY is put onto chromosome 3, where it's expressed. And now this animal, instead of making X sperm and Y sperm, it makes X sperm with SRY or without, and makes Y sperm with SRY or not, making 4 different kinds of babies. So we study 4 sexes, not 2. They're either XX or XY animals with ovaries that are lacking SRY, or they're XX or SY animals that have SRY and have testes, and therefore have male hormones. So it's a 2x2, XX versus XY on one axis and male versus female, or gonadal male versus gonadal female. And we study a phenotype. So let's say in mice, male mice, adult male mice, way more than female mice, but about 25% more. So if in this model, we can study the four sexes here and say, okay, if this sex difference in body weight is the result of hormones, then we expect the two animals with testes to weigh more than the two animals with ovaries, okay? Or if the model works out the other way, we would expect, if it's not hormones, if it's the complement of sex chromosomes, then the two XY animals will weigh more than the two XX animals, and that would be an effective sex chromosome complement. So we have, in fact, studied body weight and obesity. The intellectual leader of these studies is Karen Rui, my colleague at UCLA, who's chair of human genetics. She's an expert in obesity and metabolism. And Suki Chen and Jenny Link have been working with us on this issue. So here's a graph just of a simple phenotype, body weight. The two gonadal male groups are heavier than the two gonadal female groups. This is at 2.5 months of age. And then we say, okay, it looks like that it's sorting out that it's hormones that are doing this causing this sex difference in body weight. So the first thing you do if it's hormones is take out the gonads to see if the sex difference goes away, and it does go away. So after about a month, the males have stopped increasing body weight and the females have caught up. And after a month, there is no group difference in body weight. So that shows that it was, in fact, the hormones coming from the gonads that caused this sex difference in body weight. But if you wait another 7 months here, it turns out that the two XX animals, whether they used to have ovaries or used to have testes, are heavier than the two XY animals, showing that there's an underlying effect of sex chromosome as well as the hormonal effect. You say, well, wait a second. So these two XX animals are larger. They've always had the sex chromosomes, even when they used to have gonads. And you go back and look at the beginning here, and it turns out that even back when they had gonads, the animals that had two X chromosomes weighed about 7 or 8% more than the animals with XY in both gonadal female groups. So you have a combination of a larger hormonal effect and a smaller sex chromosome effect, which are actually in opposition, because male hormones make them bigger, but female sex chromosomes make them bigger. And so they're actually kind of canceling each other out partially, and you see the effects under different conditions. Now, this sex difference in body weight is due to the sex chromosome effect is due to fat, okay? So if you take out the gonads and wait 10 months and look at the amount of body fat, the animals with two X chromosomes are almost twice as fat as the animals with one X chromosome, although it depends on whether they used to have ovaries or testes. Going on then, so that's one model, the four core genotypes. It really shows that there's an XX versus XY difference, and the second model, which is called XY star, is another set of genotypes in which the number of X chromosomes and the presence of the absence of the Y chromosome are now independent of each other. So the animals are either gonadal males or gonadal females, and they either have one X chromosome or two X chromosomes. So the males here both have a Y, and comparing, let's say, the two columns, we're comparing animals with one X versus two X chromosome, but comparing the gonadal males to the gonadal females, it's the presence or absence of the Y chromosome. So it's a different model. And in this model, it's the same experiment. You take out the gonads when the animals are 2.5 months old. You wait a few months, and the animals with two X chromosomes weigh more than and have more body fat than the animals with one X chromosome. So this shows that this sex chromosome affects on the X chromosome. So we're making little progress here. First, we found there was a sex chromosome effect. Now we know it's on the X chromosome. And now what? What we're trying to do is find the genes on the X chromosome that are causing this effect. And we have, in fact, found two genes, but I'm not going to talk about it yet. We have found two genes that escape exon activation, which make a difference in body weight, and at least one of them in adiposity. So the real goal here is to find new genes that control metabolism in a sex-specific manner. Now, the genes escaping X activation are particularly interesting because both X chromosomes express them in females and one X chromosome expresses it in males. So constitutively, as a matter of sex chromosome complement, these genes are expressed higher in XX animals than XY, and they're particularly interesting as one class of things that cause sex chromosome effect. We've also collaborated with Mansoura Eghbali in the UCLA Department of Anesthesiologists, and she's an expert in cardiovascular disease, cardiovascular and pulmonary disease. And we found sex chromosome effects both in a cardiac ischemia reperfusion model and a model of pulmonary hypertension. So if you take the heart out of the animal and perfuse it, allow it to stabilize for a while, give it an ischemic event, which is to turn off the perfusate, the oxygenated perfusate for half an hour, and then allow it to brief perfuse for 60 minutes, you can measure how bad this ischemic event was in the different 4 groups. And we find out that the XX animals are, so here's the heartbeat, the pressure trace, and the heart's beating in this ex vivo experiment. You shut off the oxygen supply for half an hour and then allow the animal to recover. And it turns out that if the animal has 2 X chromosomes, it recovers more poorly than it has 1 X chromosome. Again, it's an X chromosome effect. This is a measure, just one of the measures of infarct size. This white area is the damaged tissue in the heart, and you can measure the infarct size and show that XX animals are worse than XY. And as I said before, is using the second model, the XY star model, comparing 1X versus 2X animals with 2 X chromosomes here, whether they have a Y chromosome or not, are worse off in their recovery than animals that have 1 X chromosome, whether they have a Y chromosome or not. So again, it's an X chromosome effect. Having a second X chromosome puts the female heart at risk here in a gonadectomized animal. Okay, again, we're focused on trying to find the X genes that do this, and our focus is on these specific genes, really only a handful of genes that are expressed higher in XX than XY because they escape X inactivation. So we are indebted to a number of NIH institutes for support of this research, including the Office for Research on Women's Health. And so thanks very much.
While the slides are being switched over, first of all, I want to thank Janine Clayton very much for inviting me to participate today. It's been an amazing set of talks so far, and I expect the rest will be fantastic. I've learned so much and gotten so excited. And I also want to thank her for the initiative because it means a lot to the world when the NIH does this, and it makes a strong statement. I also want to say that I really appreciate the lovely introduction, but I think I need to update my website because, first of all, I was the first treasurer of the OSSD, but Farida here is the current treasurer of the OSSD, so it's her problem now. And I'm now also chair of the Institute of Gender and Health Advisory Board, of which I'm quite honored to be, and it gives me an opportunity to talk about our strategic plan a little bit in this talk. But then I'd also like to say that I've modified my scientific goals a bit and decreased them to just studying oophorectomy, studying cognition in women who've had oophorectomy prior to natural menopause. So I just wanted to bring you up to date a little bit, and myself as well. So I just want to say that I wanted to call my talk and go out on a limb thinking about gender in preclinical research. So I'm taking up where Londa left off, intersecting and overlapping, and we'll see where it goes. The prelude is that, like many others, like Larry, in fact, I started off as a basic scientist. I injected single cells. I traced synaptic input. And I've moved into preclinical research, basically in humans, looking at the effects of oophorectomy on cognition in women who carry the BRCA mutation. And I can say from that vantage point, first of all, it's a lot easier if you're studying a cell. Human beings are really messy creatures. Second of all, I feel like I'm giving this talk a little bit from the perspective of somebody who's always trying to go back and forth between the animal research and the clinical case. And so I think a lot about what it is I might need to know in the modeling of preclinical research. I also want to say that it's sex and gender, because the world really writes on every body, including animal bodies. Gender isn't just self-reflective, as Londa pointed out. It has to do with your power and place in a hierarchy, which animals have. It has to do with how you're treated on the basis of your phenotype. And it has to do with environment. So in these ways, gender is relevant to preclinical models. So what I want to talk about then is a little bit what I, as a clinical researcher, would like to see or need from animal experiments to be relevant to a large extent to what I think about. And then I'd like to talk about translating gender to preclinical models, 3 examples, and then some approaches from Canada. So gender is like this psychology illusion. It's either a vase, sometimes gender and sex, sometimes it's a vase, sometimes it's a face, 2 faces facing each other, and they're always flickering in and out. That's how I see them. We sometimes separate them for purposes of an experiment, but it's really hard to dissociate them. And what we really need are models that match the human condition, because human conditions are not reductionist. Human conditions are complicated. So we need what I would call a situated science, the intersection of sex, age, time of day, all the things that we've already been talking about. And I talked about it a little bit in a paper on a chapter on a study that I did called Situated Neuroscience. We need to think a little bit about comorbid conditions, because most conditions that humans have are not just one condition. Many go together. For example, in pain, there's hardly anybody who just has temporomandibular joint problems. They usually have chronic pelvic pain that goes with it. They might even have addiction because they're being treated with opioids, et cetera. We need to think about environment, because environment affects the progression and the etiology of conditions. We need to think about social conditions for the same reason. And as Art just showed, developmental background makes a huge difference in how conditions play out. And we need to think a little bit more about whole body effects. I know I'm a neuroscientist by training, so I think about the brain. But there's a lot in the body that affects the brain. So the immune system affects the brain, the reproductive system affects the brain. And so whole body effects are going to be very important, I think, when we're making these models, at least to give a nod to the fact that we can't just study one system in isolation. So in thinking about gender in preclinical models, I think it's important to first of all situate your model. You want to think about this species, this cell type under these conditions, right? Because your species can make a huge difference in what the outcome of the experiment is. So I know we want to generalize as scientists, we want to come up with generalizable principles, but I really think it's important to be very clear about all the conditions. I would like us to think about the whole model and not just the single animals, because they're effects of a complex biology, and there's this issue of comorbid conditions. And then I'd like us to step out a little bit to think about gender in animal models, preclinical animal models, because I think that gender for rats is a little bit like memory in a sea slug. So, you know, memory is a very human phenomenon, and Eric Kandel said to himself, how can I renegotiate this human phenomenon into pieces, in a sense, to talk about it in a sea slug? And I think, in a way, we want to do the same thing with gender. It won't be directly applicable to the human condition, but there'll be many ideas that we can take from that will take us in a fruitful direction. And again, we want to think about environment as being the social world for animal models. And of course, as has been pointed out now, scientists have gender too. We have gendered expectations about what we're going to see in the material. We have expectations about what it is we should study, if it's male or female that we're studying. And also, we have a gender as well that affects the experiment. So the first example I wanted to talk about was being situated in your model. And this wasn't a groundbreaking study by any means. And it was about 100 years ago, but it's a study I did with Christina Williams and a graduate student, Philippa Miranda. And we looked at aged female dentate granule cells that were deprived of gonadal steroids long term. And we saw in females that the females deprived of estrogen long term had a decreased spine density, and the aged males did not. And it was different between the males and the females, for sure. I always tell people, you don't have to be an anatomist to see this difference. So again, in this particular species, Sprague-Dawley rats, because it doesn't work in all rats, there aren't differences in dentate granule cells. In this cell type, the dentate granule cell, we didn't look at CA1 pyramidal cells, for example. And under these conditions, this was long-term deprivation. And I'll tell you something else about the condition, which was when we gave the females estrogen back in a long-term fashion by implants of silastic tubes, we actually didn't see any difference in the dentate granule cell spine density. So the spine density didn't change just because we gave them 17-beta estradiol long-term. But when we gave them 17-beta estradiol short-term or in a bolus, we saw that the dentate granule cell spine density went up in the females, and in the males it went down. So we saw a sex difference. But we also saw that the way it was administered really makes a difference. And this kind of takes you back to the Women's Health Initiative and makes you think, okay, what kind of hormone were they delivering? How was it being delivered? How old were the participants? It makes you need to situate what the outcomes of the studies were in terms of the details of the studies themselves. And I think that's important the important insight preclinical models can give us. Then I think we can also think about the whole model and not just the single animal. So what is your model that you're looking at? So Jane Danska, in this Markle et al. Science paper, which I'm sure many of you know about very well, Jane Danska asked about type 1 diabetes. Why is it more common in women than men? And one of the things that she noticed in her animals was that when males and females were raised in pathogen-specific, pathogen-free specific environments, the males had a very different microbiome than the females did. Yes. And when they were raised in a germ-free environment, there wasn't a difference. So the environment made a big difference in what was seen. And then she also saw that in the specific pathogen-free environment, testosterone, and between germ-free and specific pathogen-free environment, testosterone levels were different in both sets of animals and in both between the environments. So she saw this correspondence now between testosterone levels and the microbiome, which is really pretty incredible. And so she went on to look at immune system function and the model for type 1 diabetes, and found that the males were actually protected if they were raised in this specific pathogen-free environment, whereas the females were not. So she concluded that the higher testosterone made a difference. If she transplanted, do I have a slide? Yeah, if she transplanted the male microbiome to the females, she actually found a decrease in the incidence of type 1 diabetes. So this is a pretty amazing experiment. It's very complicated, but it is what it is to be in a complex human world, and it's an animal model. Another animal model, going to the metaphor, using environment instead of, for example, self-reflective notions of gender, was an old experiment done by, out of Merznick's lab. Jerry and Judith Stern, probably was Judith Stern's idea, to look at the representation of the ventrum in the somatosensory cortex of lactating female rodents. And what they found was that in the females who were not lactating, the representation of the ventrum was the same size as, well, it was quite small. And in the lactating female, the ventrum was increased at least 2 times. Now, this is a really interesting experiment because, first of all, it speaks to a particular female condition of pregnancy and lactation. And it tells you about the experience of lactation for females. And it makes you begin to wonder, well, how do hormones play into this? How does the hormonal change after pregnancy and in lactation? Does it potentiate this expansion? Does it reduce the expansion? I mean, does it influence the expansion at all? So it opens up new questions, and it also makes you wonder, what's it like for human females? What's our somatosensory cortex like? And it turns out that there actually is not a model. There's not a map of the female body in the female brain. So Penfield mapped the male brain, but he did map the female brain. He mapped about 10 women. He only reported on 1, and there actually is no map that was generated. So it raises all kinds of interesting questions, but also shows that if you take the environment as a metaphor for the gendered world, you begin to learn things and raise questions about being gendered female. Now, this is the experiment that everybody's been talking about already, now at least twice, thinking about gender in preclinical models where scientists have a gender too. And it's just the amazing experiment coming out of Jeff Mogul's lab showing that when there was just a male researcher sitting in the room, the pain thresholds of rodents was lower, male rodents was lower than when female observer was sitting in the room. And this he correlated with the increase in stress hormones in the males, which then reduced the pain threshold. So this is another complicated, imbricated kind of model that also talks about, well, really what philosophers would call the second-person interaction. It's almost, the interaction between the environment and the subject. And it's something that I think is really important to think about now. I don't think we can any longer, talk about the model as existing in isolation from the investigator. So to talk a little bit about approaches from Canada, first of all, Londa already raised the point that in Canada we have had a mandatory reporting of sex and gender questions in our grant applications since 2010, which does include preclinical animals, preclinical models as well as humans. And how successful or not successful that's been has had a report. It's been written up. And what we're hoping to take it to now is the level of actually having this question count in the review process. And we'll see how successful we are, but the director of our institute, Joy Johnson, is working really hard on having this become a part of the review process. We also have a strategic plan, 2017, going forward, which is really our key goal is to bring the social together with the biological. And while at CIHR, if you look at the total funding for biomedical grants as opposed to sort of psychosocial grants, the money is really going into the biomedical grants there. What's the kind of funding that IGH, the Institute of Gender and Health, really looks at is the kind that is integrating the psychosocial, and what we need to do now is to bring the biomedical into what the Institute of Gender and Health is already very strong in. We also had an IGH-sponsored workshop in 2013 for bench scientists on the inclusion of gender and sex, which resulted in Stacy Ritz's paper, which has already been cited here. So there are things that have been being done at the Institute of Gender and Health. Then we have an emphasis on something we call capacity building in Canada, so training. And we have had for 3 years an Institute of Gender and Health, Summer Institute for graduate students and postdocs that tries to bring the biological together with the social in trying to unite sex and gender. And we brought a lot of qualitative interviews, a lot of qualitative work together with more quantitative work and tried to help students think about how you would do that. There's a specific strategic training initiative coming out of the University of British Columbia called IMPART, which is looking at the effects of sex and gender on addictions. And it also has really worked hard at bringing students from the biomedical and quantitative sciences together with those doing more qualitative research. And the training program includes monthly WebEx seminars where people really begin to integrate what they're doing. And then there's the collaborative graduate program in women's health. It is indeed a graduate program that tries to bring together sex and gender. We have 2 key elements in this program. It doesn't affect every graduate student, but it affects those who are interested in women's health or those who are interested in doing this kind of multidisciplinary research. And it's been going since 2007. Our 2 key elements are a course called Gender and Health and a student seminar series which runs every single month. The goal of both of these is to juxtapose the biomedical with the theoretical. So in the course, whenever we read about, say, autoimmune diseases being more common in women than men, and the students read Eleanor Fish's paper on autoimmunity, sex differences in autoimmunity, they also read Donna Haraway's paper on autoimmunity, which is an extremely interesting paper. And we, in the seminar, we also have faculty from different disciplines respond to the students' work. So the last thing I want to tell you about is the Canadian Consortium on Neurodegeneration and Aging, a brand new initiative out of Canada. Pan-Canadian, PI Howard Chertkow out of McGill. We have 3 themes, 20 teams, 8 platforms, 4 cross-cutting programs. And myself and Mary Tierney proposed a cross-cutting program on women in dementia. And so we are now faced with the task of integrating gender and sex across all of these platforms, teams, and themes having to do with dementia. And it will be exciting. We are just now developing our principles and procedures for this initiative, and I'll be happy to share them with you later. Anyway, in thinking about gender in preclinical models, best practices. Are the same conditions different for females and males? We should get rid of this notion either or, binaries, okay? This has already come up today. We should talk about overlap and pluralities. We need to provide individual data points so people can actually see the overlap. We need to acknowledge the particularities of our tissues, cells, and bodies, recognize difference without reifying difference. Be open to similarities, be circumspect in interpretation, and determine which differences matter. We really need to do a piece of PR teaching people that difference is just a difference. It's not better or worse. And I just want to say sex and gender matter, and to quote the IGH 2017 strategic plan, have you considered the possibilities? Thank you very much.
So at this point, I'd like to invite the 3 speakers as well as our keynote speaker up. And the floor is now open for questions to all of these speakers as well as comments.
You guys did a wonderful job, each and every one of you. I just want to make one comment, Londa. So the NIH inclusion policy applies to all NIH-defined clinical research, not just phase 3 clinical research. The 1993 one applies to all NIH-funded clinical research, not just phase 3 clinical trials. Okay, so I just wanted to correct that. There are additional requirements for phase 3 clinical trials. In addition to women being included, studies have to be designed such that a valid analysis for sex difference can be performed. So that's the additional requirement for phase 3 clinical trials. Okay.
Jillian, could you speak to how Canada manages for the basic sciences, all model organisms, so mouse and rats, monkeys, maybe fairly obvious, but what are the mandates or the suggestions, recommendations for Canadian basic scientists using zebrafish or using xenopus or other model organisms, C. elegans? What's been the thinking in Canada?
Okay. Yeah. Okay. That's a great question. And whenever somebody says it's a great question, of course, it means they don't have an answer. We put the policy out there on the grant applications, wanting people to answer it. There are training materials that are being developed, and there are some written documents that Joy Johnson herself has authored, but I will say they don't go into the granularity of how you do this at the level of cells and non-mammalian animal models. Again, I think we push the idea that people need to think about this and metaphorize and imagine, and I think they do, but there's no actual policy on it.
You metaphorize. Do you think of animals that are mammals? Is that the metaphorization boundary condition? Maybe to Londa as well. How far do we metaphorize given the two definitions that you've provided us, which have to do with environmental interactions? And so you can say zebrafish have this, where are the metaphoritic boundary conditions?
Well,
located, I think.
Yeah, well, you want to take...
No, that's it for me.
Well, I just want to say that, first of all, there are fish that definitely have genders that change. So the protogynous fish, their sex changes, but there must be something in the interaction of the fact that there is no male.
They can detect there's no male, but that doesn't mean they have gender.
Well, I really think that they have gender in the sense that there is an environment in which they're
depends on your
interacting.
The way the definitional interaction with the environment, if that is gender. I think that's a good place for
the sex of the fish that changes. It's the sex that changes in reaction to the gender interaction of the group.
Right.
That's what I would say.
Yes. Thank you.
Finally, could I press Art on this topic as well? So sex chromosomes and parts of sex chromosomes begin to matter to the biology as we go up toward human. But I wonder if you and your colleagues have thought about, alligator sex is imposed by temperature. So maybe you're identifying genes that are going to ultimately be those temperature-regulated genes. Or how do you think, maybe in this context of sex chromosomes or parts of chromosomes as they reflect what we're now metaphorically calling sex, SRY, in the case of the 4 core. How do we broaden that thinking?
Well, first of all, I think research on non-mammals is enormously informative, especially when you get into the evolution of genomes and the sex-specific evolution of genomes. So I spent a lot of time working on birds in our research, and I have and just want to say that I got more bright ideas from studying birds than I ever did from studying mammals, including the idea that the sex chromosomes might make a difference. Now that's just me, but the whole concept of the evolution of sex chromosomes and this dosage compensation issue, which is a major part of evolving sex differences. If you look beyond the mammals, you see different patterns of dosage compensation and how genomes handle sex differences in gene dose. Okay, and birds actually don't, at some level, don't look like they handle it well, but they're doing just fine, and that raises a lot of questions about dosage compensation. So, I think there's a place for this kind of non-mammalian research. Of course, we spend most of our money on mammalian research because we think we can model diseases much better in a mouse than in an alligator, but that's partly because we know a hell of a lot more about mice and we can genetically manipulate them. But I'm a big fan of comparative research because it helps us define, gives us a real place to stand to view the mammals, including ourselves, and kind of see how mammals differ and why do they differ and how did it evolve. So I don't know if that answers your question, but I at least could put in a plug for birds. Yeah. Well, yeah, and then there's all the endocrine disruptors. The sentinel animals are often non-mammalian, so they're quite important.
So I have a question for Dr. Douglas. But is the EPA starting to understand that maybe sex differences in pharmacokinetics and pharmacodynamics are really but they're large, and that I was just concerned when I saw the immunotoxicity. That is one of the categories where there's no, you could use either sex. And then also I had a question about, you had this table where gender differences, sex differences was one of the components that you were looking at, but you also had it at the same level as the effect of life, where you are in your lifespan, or I'm just wondering, is the sex differences kind of going down the chart of all the different things you're looking at, or is it only at one stage, say, for example, a lifespan?
Well, I think I think sex differences are generally evaluated in the guidelines, the EPA guidelines, and so it's just a standard component actually of the testing that's done, rather than thinking of it, outside as well. Should we do this comparison or not? Really, it's the exceptions. There are a few exceptions where it's not looked at. And but I think in general, the major tox testing that's done to screen a chemical includes both sexes.
I have just a general comment that comes out of hearing a lot of the lectures today. Quite often, we are reminded that when it comes to sex influences, there is overlap, and it's presented. I've learned in almost an apologetic kind of way. There's overlap. Don't worry, there's overlap. And whenever you present your results of your sex influences, you quite often have someone say, but it's not true for every man. It's not true for every woman. And I just wanted to take the opportunity to say that comment makes no sense. There's overlap between schizophrenics and controls. There's overlap between Alzheimer's patients and controls. The fact that there's overlap means nothing about the general importance of the sex influence issue, and I just wanted to say that when people say this to me, I usually ask them, do you think aspirin reduces pain? And they say yes, and I say, what? You think all doses of aspirin reduce all kinds of pain equally in all people at all times, no matter the pain? Well, no, I never said that. But you said aspirin reduces pain. So you see, there's this strange thing that I just wanted to say seems to apply selectively or disproportionately to the sex influence issue, where you have to somehow be apologetic about the fact that the nature of the effects are just like those in the entire rest of biology. This is not so much a question as a tirade, but I just felt the need to comment on that, because it just came up here before this thing is all over. I'm done.
I'd like to say something about that.
Well, I'm going to tirade right back at him.
Okay,
because I say the same thing.
See if you say the same
I'm going to say that only a man could say what you just said.
Oh, wow.
Because only a man doesn't know the discrimination that women have put up with in every realm of life for so long, and how we are so trained, first of all, to apologize for anything, but also how we have to be so careful about sex differences research being abused and misused and misinterpreted, and to explain why a woman shouldn't be president, and because they have those hormones, and because they're emotional and weak, and et cetera, et cetera, et cetera. And also, I would like to say that being a woman is not being a schizophrenic, it's not having Alzheimer's disease, it's not having a pain, it's a biological category. So your argument doesn't hold.
I just thought.
It doesn't diminish it. It accurately reflects it. No, it doesn't. You are such an extremist, Larry. You are a convert. That's your problem. You are an extremist as a convert. Excuse me, Patty. It's a perfectly legitimate
I would like to say that when I say there's overlap, I don't say it as apology. I say it as actually an interesting finding. Because, for example, I've known men who have cardiologists who have had heart attacks in the same way that women present with heart attacks, and they haven't recognized it in themselves. And as a result, in fact, they have died. Okay, so that in that case, it's really important to understand that women don't all get heart attacks in one way, and men don't all get heart attacks in another way. I think in the clinical realm, so I don't think I wouldn't say it apologetically at all. I think it's actually an interesting fact and needs to be acknowledged. At which point we will come to fisticuffs.
For those of you viewing online, we'll reconvene at 2:45.
Good afternoon. My name is Lee Alakel, and I'm from the National Center for Complementary and Alternative Medicine. And you might ask, what is someone from NCAM doing at this meeting? And yes, we do a lot of human-based research, and I was a human researcher for many years. But I'm very interested in this issue of sex differences, and I'm extremely interested in women's health. So, and I think there's a lot that those of us from the clinical arena can learn from basic mechanistic work, and vice versa, as we have seen today. So I think that this crosstalk is really, really wonderful. It's excellent. So I'm going to just outline what the purpose is and provide some of the questions today. This session is Cultivating a Culture of Sex Matters Across Multiple Disciplines. And the purpose is threefold. One is to discuss when sex matters and when the biological variable of sex should be considered in science. And two, to determine where gender fits into the research realm. And three, to determine if there is a research space for single studies and if such studies will result in no harm. Now, I have some slides planned. I may not go through these. I may go through them very quickly and skip through them just in the sake of time, because I really would like to turn it over to our speakers. But this first, to discuss when sex matters and when the biological variable of sex should be considered in science. And I would say that sex should always be considered in scientific research. Always, always, always. It's not that you always will contrast males versus females, but you always have to consider it. And so I have a quote here from one of Dr. Milner's articles from the American Physiological Society, and I'll leave that to you to read yourself. But essentially, this is just it just demonstrates how important it is. And so we've heard this many times today. Sex chromosomes are in every cell, and they certainly play non-gonadal functions at sex hormones and sex differences, as well as in terms of gonadal function. And so we know that this is important, and I won't belabor the point. And so again, sex should always be considered in scientific research. And I could give you lots of different examples, but I think I will leave that to our speakers. I want to give you one example that was raised at this North American Menopause Society meetings that I just attended. There was a whole session for one day. It was a pre-session on thermoregulatory responses, and it was on the basic underlying mechanisms. It was absolutely fascinating. And so one of the issues that was raised was, why do we only study this in women? Why do we only study vasomotor symptom instability in women? Really, men have this as well with gonadal hormone deficiency. So I think that the time has come for us to really begin to think about some of the basic underlying mechanisms that we've thought, well, we should only study it in women. And I think the other example was with osteoporosis, and we need to study osteoporosis in both men and women. I think it would be very illustrative. I will say that, certainly we know there are differences in thermoregulation between men and women. For example, we women typically have, particularly postmenopausally, a much more narrow thermoneutral zone. So men have a much wider thermoneutral zone, meaning that they can withstand temperature fluctuations much better than we can. My husband reminds me of this all the time. So I just think that there's a lot to learn. And I've put up a couple of examples here. But again, I'm not going to belabor this because I think that our speakers will do this for us. We've already heard about where does gender fit into the research paradigm. And this just really just reiterates what we've already been talking about as far as the difference between gender and sex. And we know what that is. And so, we've already talked about a lot of this. This is the liability of going in the afternoon, but it's also the advantage because you can move pretty quickly. And so I think that I want to make the point that it really depends upon the questions being asked. It depends upon your experimental design. And each investigator has to decide, how am I going to include sex in my research paradigm? So I think that that's a very important question. And we're going to answer all of these questions today, mind you. So certainly when we're thinking about space for single-sex studies, certainly with pregnancy, a single-sex study might be appropriate. And there are other really good examples. And in terms of non-reproductive function, it really depends upon the question being asked. And I'm just providing these as some examples here for you today. And those are the references. And now I would like to welcome Dr. Miller, who is no stranger to this group, I know. And I just want to say a few things about her. She's a professor of physiology and surgery, also a research director in the Building Interdisciplinary Research Careers in Women's Health at Mayo Clinic. And I want to just mention a couple of things about her. Her current research focus is on preeclampsia of pregnancy and menopause, as well as examining sex steroid effects and changes on accelerating the atherosclerotic process in cardiovascular disease. Now, I think for the sake of time, I'll do both introductions at once, and then I'll turn it over to our speakers. And so the questions that Dr. Miller has been charged with, although I understand you may have questions of your own, but working across disease states, what role does sex play, and how do we think about sex with regard to disease comorbidities? And also, we'd like to welcome Dr. Catherine Sandberg, who's at Georgetown. She's the director of the Center for the Study of Sex Differences in Health, Aging, and Disease, and a professor of nephrology and hypertension in the Department of Medicine, as well as a PhD program director. So we have questions for Dr. Sandberg. When approaching a new research question, what methodology can be used to determine whether or not sex should be considered? And are there circumstances when you can justify studying a single sex? And so I will just say a couple of things about Dr. Sandberg's research. And it is also in the area of gonadal steroid regulation, particularly of the renin-angiotensin system, so cardiovascular disease and health. So their research areas actually seem to overlap a bit. So without saying any more, I will turn this over once we get up the slides. Okay, which ones are yours?
Revision 2, Miller.
Okay, there we go.
Okay, great.
Excellent.
Okay.
Thank you so much. I am so excited to be here today, and this has been such a stimulating conference. Thank you, Janine, and the Office of Research on Women's Health for inviting me. I'd like to acknowledge our funding from the Office of Research on Women's Health through the SCORE program, which is co-funded by the National Institutes of Aging. And I wouldn't be here today, I don't think, if I would have participated in this particular activity. As Lee mentioned, I was charged with discussing cultivating a culture of sex matters across multiple disciplines. And I'm going to do that today by addressing three questions that I was kind of charged with. Is it acceptable to exclude sex as a variable in experimental design? I turned it around a little bit. Can a specific criteria be developed for excluding consideration of sex in experimental design? And what tools can be acquired to work across disciplines toward understanding animal models of complex human disease? And I think this is really key, and it's been brought up a couple of times today, but I hope to leave you with some really definitive questions that you might be able to ask to help direct the answer to your own research. So the first question is it acceptable to exclude sex as a variable in experimental design? And I think you already know the answer to that. Lee answered it, and I'm going to answer it again, as every cell has a sex. So the fact of life is sex is always a biological variable in animal-derived material. Now, I put animal-derived material here, and I should have put vertebrates because then we start talking about amoebas and paramecium and things like that, and I don't want to go there. I don't study those. But when you're talking about a vertebrate animal, you've always for the most part, you always have sex as a variable, and it needs to be identified. It needs to be identified in the methods. When I was in graduate school, we were taught, write the methods so somebody can reproduce your experiment, which is the details. And I think the whole impact factor thing has really deterred us from that, because typically the methods get shortened, they get put into the supplemental material, and who knows what everybody has done. We have to get back to being precise in science, telling each other the exact parameters of our experimental design. Sex is key to that. So, second question, can specific criteria be developed for excluding consideration of sex in an experimental design? So I've already told you that we have to think of it as a variable. But this leads us to the question is it appropriate to study have single-sex experiments, and does it do no harm? I was kind of put off a little bit by the do no harm thing because I think when I frame this a little differently, you'll see that it's not an issue, okay? So when we think about sex differences, we think of, I used to say 3 categories, but I've lumped them now into 2 categories. First of all, you have to consider that when we talk about sex, it affects every system of the body. So we have to get back to thinking integrated physiology if we're going to really advance preclinical studies to the translational level. Then we can think about it in 2 ways. We can think about it as sex-specific versus sex-different, okay? So what are sex-specific conditions? These are conditions which are unique to one sex. Now, clearly, we can't study prostate cancer, testicular dysfunction, ovarian or uterine things in the opposite sex because the organs just aren't there. So that takes care of a whole group of studies which have to be sex-specific in order to understand the mechanisms of the diseases associated with those organ systems. The second set, then, because we're thinking as integrative physiologists, are conditions or diseases which occur more frequently in one sex or the other, present differently in one sex compared to the other. For those of you who are physicians in here, think about one disease that doesn't fit the second category. I don't think there is one. So when you break it up into these 2 separate categories, whether it's sex-specific or sex-different in terms of presentation, morbidity, mortality, you have to think that it fits into the realm of you have to study the sex differences. Now, I'm going to use Art Arnold's special phrase of sex biasing factors across the lifespan. I love this picture for a variety of reasons. First of all, I think it's just very sweet that it shows the life stages, but particularly, I want you to focus on our woman right here. The female body is designed
to support pregnancy. Okay? So pregnancy comes and goes. Fortunately, it's not a permanent state. And so you have to accept, if the female body can adapt to this condition, there has to be underlying physiological mechanisms that allow that adaptation. And what influences that adaptation? Is it the sex chromosomes, or the hormones, or is it a combination of those across the lifespan? And others today have provided significant evidence to say that it's a combination of the two. You may start off with sex biasing factors with our chromosomal dictated, but that's going to change across the lifespan as that hormonal environment modifies. So when we talk about parameters that are going to influence our conditions, we can talk about sex, we can talk about gender, which I'm not going to really say, but critically we have to talk about hormonal status and age. So when we think of these key variables that are going to affect the outcome of the experiment, the sex differences may depend on whether you have the appropriate age and the appropriate hormonal environment. So we're really thinking about this in an integrated way. The sex biasing factors across the lifespan, one of which is sex chromosomes underlying them all. So the sex-specific conditions, I would say these are pretty much reproductive and neurological organs or processes. And I didn't know what to do with established cell lines here because they do have a sex. And, I think you should identify the sex in your paper. I think it should not be a given that everybody understands that certain cell lines come from a particular source. We should say that. And I think we're at the stage of scientific investigation where we should say that there are data needed from cell lines of previously unrepresented sex. So if the cell line has to, a lot of information has come from those. I'm not going to try to diminish any of those discoveries or observations. But this is a time to say, well, what would happen if we used a cancer cell derived from somebody else, from a male? Or a pulmonary carcinoma from another cell, or whatever particular cell lines it may be. I think we need to start exploring the other sex. So what about the sex differences? How can you use what we know about the underlying physiology that is going to be different across the lifespan to help you design your experiment? So I'm thinking about this, and I would like to propose these questions for the investigator to tick off in their head when they start to write their application and design their experiment. The first one is there evidence of a sex difference in the disease, incidence, prevalence, morbidity, or mortality in humans? And if so, what is it? If we are to be translating our findings to the human condition and improving human health, we need to understand what that condition is in humans first. Many basic scientists do not. And this may help them get started thinking. And we have to keep in mind, this is brought up many times today, that if there isn't a difference in the literature, you have to ask your question, has it been studied? And, or if there is a difference and it has been studied, has it been reported? So these are sort of conditional questions that you have to put on that to say, well, just because it's not in the literature doesn't mean there must be no difference. Because if you look at the clinical outcome data, you're almost for sure going to see sex differences in any of the diseases that you look at, either prevalence, incidence, morbidity, or mortality, or treatment options. Second, I think you need to think, is there an experimental model of the condition or the disease? Now, we have a lot of experimental models. I don't like the word model, but, experimental animal to investigate what you want. But I think more importantly, we have to analyze whether that actually represents in an accurate way the condition which is observed in humans. And this has again come up several times today. I had one of our Birch scholars applied for the condition for their thing, and she was studying osteoarthritis, and she's using a male model of disease. And I said, well, osteoarthritis, there's a female prevalence. Have you ever considered using the females in any of your experiments? And her answer was no. And I'm thinking, okay, this is a interdisciplinary research program in women's health, and you think you prepare a little better for the interview, and but Anyway, she's not in our program. but I mean, this is what we have to change the thinking of whether the model is actually going to tell us something about the human condition. And finally, are data lacking from an experimental model in one sex compared to the other? Now, this has been brought up, that we need to have equal numbers of males and females in experiments going forward. And I will put forward the fact that we have a huge body of data on male animals. It's pervasive in the literature, okay? We don't know if there's sex differences. Should we require every investigator going forward to look at that exact same condition when it's already been published? So I think even in those physiological systems where we know it might be different between men and women, there would be a place for single-sex studies if they are providing a database which is missing, which is information about that sex. And, we see meta-analysis all the time of literature data. If you're looking at the same kind of experimental design, as much as you can get out of the methods that may not be adequate, and you're designing that experiment in the opposite sex, that data should have some value and actually be able to formulate or inform further hypothesis generation in terms of investigating the mechanisms of that sex difference. So the third question is, what tools can be acquired to work across disciplines towards understanding animal models of complex diseases? I was trained as an integrative physiologist, so this is kind of an easy way for me to think, but not everyone has been. And I was also trained in the idea you have the question, you develop the tools to get the answer, not the other way around. So I think what we have to put forward is to encourage people to think differently about how they design their experiments. Not only how they design their experiments, but how they develop their teams and who they work with. Okay, we hear this all the time at Mayo, think differently because we've got to save money and adjust to the Healthcare Reform Act and so on and so on. We have to think differently out of the box, think differently, think differently. I'm thinking, well, why can't we think differently in research as well? And we can. So here's some things to consider. Do we work in silos or networks? Okay. Do we have limited resources or do we share those resources? And how can we, if we have limited resources, how can we better share them? Not necessarily, even within your laboratory or the lab down the hall. And can we take a more programmatic approach to our ones that are individually investigator-driven? And I would think that this is sort of a way to really think out of the box in terms of we want people to do translational research, we want people to do interdisciplinary research, but we still fund the single PI program. Within that paradigm, can we think of it differently? Now, we have multiple PI kinds of grants and so on, but is there another model that can be developed? So, for example, in the SCORE projects, we have to have, human component and molecular animal-based component and so on. And I've used this from our own study of how we look at either a sex-specific conditions of pregnancy and menopause. But I think we can also design this approach more broadly. Think of the sex-specific or sex difference condition that you want to study. Can you study it in terms of the whole animal or whole individual? Other molecular cellular mechanisms that could be obtained from that same particular animal model applied to really understanding what's going on in the organ, the structure in terms of function. So if we take this question here, and even when you do clinical trials, when we did the KEEPS trial, although the question related to cardiovascular effects of the hormones in early menopausal women, we designed 8 different ancillary studies associated with those women without extended burden to obtain a variety of information. So our resource, our volunteer, was actually a pooled resource for several investigators. When I first went to Mayo, we did a lot of our vascular work on dogs. Dogs were expensive. We had a pool dog, okay? We called around to everybody that was interested in doing some vessel or brain or eye or ear or whatever. We pooled those resources and said, if we buy the dog this week, will you buy the dog next week, and so on and so on. We didn't have to put together some NIH thing or a SCORE project. it were investigators saying, look, I only have this amount of money. I need to have this animal. Do you want to share it? I think we'd have to encourage sharing of these limited resources. So in this way of thinking, we can see how easily a shared resource, like an experimental animal or within an institution, could be shared for physiological studies, molecular testing, other kinds of conditions or other organs that might be available, and really develop a network. Okay? Now, what's limiting some of this? Well, I think the tools have to be different. We have to think network instead of silos. Now, I'm going to get in a little bit of trouble for this, but I'm going to hope Larry will bail me out, because he initially started the program talking about brain networks and connectivity in terms of more sort of matrices and sort of the male brain and networks in the female brain. And I'm thinking, do we, and Art brought this up too, would same data be interpreted the same way between a man and a woman, depending on their point of view and how they process that information? Not that it's better or worse, that it's just different, and perhaps it's time to think more network than matrices in terms of cross-disciplinary things, which I see matrices all the time at Mayo in terms of programs. The next thing we need to think about is how can we really maximize our limited resources, but clearly without compromising precision. And we, as scientists, want precision, we want control, we want to control those variables. We have to be able to think how we can share these resources without compromising precision. And finally, I think we need programmatic approaches within the individual R1 laboratories and really think creatively. And I think I'd like to challenge NIH about this, is alternative funding models for how we do some of these mechanistic work of sex differences. You know, NIH has been around since it's almost as old as me the review process, and it hasn't changed a whole lot over that time. And I think it's time to maybe take a deep breath and say, in this new world, we heard today about the Cancer Society or Cancer Institute sharing databases on the cloud. Is there some way that we can do that also with the animal resources? So I'm going to reiterate the questions and close today and leave you with the answers so that you can go off and be successful. Is it acceptable to exclude sex as a variable in experimental design? And I'm going to say no. Lee said that too. We have to, for all experiments using vertebrates, sex is always a reportable variable. Okay? So we have to report it. Can sex-specific criteria be developed for excluding consideration of sex in experimental design? And I think is here where you have to really consider, is it sex-specific or sex-different, and what are the characteristics that you can, or the criteria that you can apply to those excluding things. And most importantly, can the model be justified on the basis of disease or outcomes in humans? And I think depending on what the answer to these two questions might be, or to these two considerations, you will get the answer to should you even develop specific criteria rather than having justification for what you are actually doing. And finally, what tools can be acquired to work across disciplines to understanding animal models of complex diseases? And I think that you have to think differently in terms of network, resource sparing, and alternative funding models. And with that, I will leave you to go forth and develop your interdisciplinary programs. Thanks very much.
I don't know if we have time for one question, just one. We'll have time after, at the end, but yes.
I'm just curious. Is this on? No. Hello? I'll speak loudly. I'm just curious on the question about models.
Mm-hmm.
There are situations right now in which dominant models are used to study disorders with sex differences in their incidence, but often situations happen where, for example, I'm thinking of irritable bowel syndrome, something that disproportionately affects women. Some of the dominant models used in that domain work great in male and simply don't work in female animals, and everybody in that domain knows it.
Mm-hmm.
Isn't it time to take that as low-hanging fruit and simply start to say that is unacceptable?
Yes. Yes, absolutely. And that's just directly to my point. We have all these data out there in terms of what's going on with irritable bowel syndrome in male animals. Let's study the females, okay? Is that justification for studying single-sex study? Yes. Is that an example of the animal model not recapitulating what might happen in women or in the human population? Absolutely.
I'm saying it's worse than that. There's situations where they know it works in the male animal and not the female animal, and they continue to use it to model irritable bowel syndrome. How is that acceptable?
To me, it's not. Okay, you bring it up at study section when the program comes up, and people around the table go, "Oh, there's that sex person again talking." Peg can probably relate to that, but it's not. I don't think it's acceptable. It's not defensible, and I've heard people say that if we're not treating our women patients with the cutting edge, or the men with that procedure that will work, it's unethical. Okay. On that note, I'll turn it over to Catherine.
And I'll address that question through my talk. So it's a real pleasure to be here today, and thank you very much, Dr. Clayton, for putting this together, and for Leah for organizing at least our session. It's just fantastic. And what I thought I would do, since I'm the last talk in this session, is not talk about what Virginia's just talked about. As you know, the session purpose already, to discuss when sex matters and when the biological variable of sex should be considered in science. And I think I don't need to say what it's already been said. Yes, it matters. And determine when gender fits into the research realm, and Gillian Einstein gave an excellent talk on that. So, and the third topic, which is the last topic, which is why I'm going to talk about something else, is to determine if there's research space for single-sex studies and if such studies will result in no harm. So I thought I'd give you a couple examples from my own research and to make my case. So first of all, I want to point out that there's this article in, is there a pointer here? Yeah. No, a mouse, okay. So there's an article that's written by Guzik and David Harrison's lab that came out in 2007 in the Journal of Experimental Medicine on the role of the T cell in the genesis of angiotensin-induced hypertension and vascular dysfunction. And you can see by looking here at the total downloads that it's a very popular article and a Science Citation Classic by now, and a paper that in my field caused a lot of labs to start studying immune modulation of hypertension. But nowhere in this whole article did they mention the sex of the animals. And I searched, and this is what they showed. But there was no mention of the sex of the animals. So the y-axis is blood pressure, and the x-axis is days. And this is a common model in my field. We infuse angiotensin's peptide, increases blood pressure, and it's a model of hypertension because it's a good model of hypertension in that clinically we treat hypertension with inhibitors of the synthesis of this peptide or inhibitors of the receptor it binds to. So you can see when you infuse angiotensin, blood pressure goes up. That's the black line. It's just a regular mouse. And then in a mouse that has no T cells, that's the purple line, the blood pressure doesn't go up as much. And so this is a first indication that the immune system is playing a role. Without T cells, blood pressure is lower. And then when you give back T cells, it's a green line, you can restore the magnitude of the hypertension. So the authors concluded that the T cells are contributing to hypertension, and so there's immune modulation of hypertension. Now, I told you there was no indication of the sex of the animals or the T cells in this paper whatsoever. So if you tried to reproduce this study, why did it do that? What happened? Why is it going away? I hope that's not going to be a problem. Okay. Well, that's a shame. I'll just have to tell you. So if you reproduce this study and you use male T cells and you do adoptive transfer, you will restore the hypertension in the male mouse. And so if you want to reproduce this study and you happen to choose males, you'd be successful and you wouldn't waste money and time and resources. And you would say, yes, it's that finding, I can reproduce it and I can go on and study the mechanism. But if you used female T cells in the same male mouse, you would not reproduce the hypertension. In fact, if you used the CD4s or the CD8 T cells, you'd actually lower the blood pressure. So that if you give female T cells, you cannot drive the hypertension, and if anything, you protect the mouse from the hypertension. So if you're thinking about reproducibility, here's a classic example. If somebody had tried to do this whole study in females, they would not have been able to reproduce these findings. And they would have wasted a lot of resources. So the graph you can't see, which I don't know why, must be an anti-Mac thing, would show you that we can reproduce this study if we use male T cells, but not if we use female T cells. The only difference in the hypertensive mouse and the normotensive mouse is the sex of the T cell. So here's another example. This is another model of hypertension. It's where you take out the right kidney, and then in the left kidney, you tie in kind of a figure-8 configuration the kidney so that you cause constriction and damage the kidney. Now, if you look at the male kidney after about 6 to 8 weeks, you see a lot of damage to the kidney. You see a tremendous amount of glomerulosclerosis. But if you look at the female, there's no pathology. So here are two examples of how I would say it would be relevant to study the male but not the female, because if I'm going to look at the mechanism of this renal RAP-induced hypertension and then what's causing it, there's no point for me to include the females because I can't get any pathology. So I would just be including animals, going along, looking at mechanisms, and I would learn nothing except I would just be wasting money, because the female in this model does not develop the renal pathology or the hypertension. And the same with in the model of angiotensin-induced hypertension. If I'm looking at immune modulation of the hypertension, there's no point for me to use the female model because there's no immune modulation of hypertension. But women get chronic kidney disease, and women get immune-mediated hypertension. So we need to study those conditions. We just need the models. Now, what I can say is that these models are good models to look at resilience, not susceptibility to disease, but resilience. And so in that case, we're going to try to figure out what's protecting the female. And the design of those studies is going to be inherently different than looking at what's causing the susceptibility. And so I think this is a good argument for why these studies should be separate, but conducted in both sexes. Study the susceptibility in one and the resilience in another. And so that's why I would recommend that NIH balance their portfolio by having models, if there aren't models available to study chronic kidney disease, put out RFAs to get investigators to come up with new models for looking at chronic kidney disease in females, or to at least appreciate the fact that this is a great model for looking at resilience to renal RAP-induced hypertension, and the same with the immune modulation of hypertension. So here's an example of a counter, and this is my colleague Mike Ryan at the University of Mississippi, and he's interested in lupus, and he's interested in the hypertension that's associated with this autoimmune disease. And you can see in these mice, the blood pressure goes up when they develop the lupus. This is the NZB-WF1 mouse. So they get high blood pressure. You can see this black bar, and if you compare that to the control. And this is true for the female. There's no point for him to include a male in this model. So there's no point for him to study the male mouse because the male mouse does not develop any of the lupus-type pathology until the very end of life. And so there's no point, and it would be a waste of dollars. However, men do get lupus, and I think that justifies that we study lupus in male animal models of disease as well. So I think there is space to do single-sex studies, and you can think of it in terms of susceptibility or resilience to disease, and those are good reasons. But I think that we have to be careful because this could be turned around and used as an argument for why business as usual, because there are no good models of chronic kidney disease in females, and immune modulation of hypertension in the female is also, we don't have good models for that either. So I think we have to be careful in how we look at this, but I do think that there is a need to do single-sex studies because these are very different mechanisms, susceptibility and resilience. So I thought since I have a little bit more time, I would really go back to what the session title is, and that is, how do we cultivate a culture of sex matters? Well, I like this. I was looking for something comic because it's the end of the day, and I thought we all could use a laugh. So I saw this little comic strip, and I thought you might enjoy it. So we must change our culture of cynicism and negativism. You too will be the happiness committee. Come up with some ideas to improve morale. Okay, so far we've got raises, slap the boss day, and nude Fridays. I feel cynicism melting away already. So I use this as a way to talk about how do we change culture. And in the business world, they are thinking about this, and we don't do it as much in science. And here is an image of how we see culture change, that it's just a tremendous activation energy to change the culture. But we can change the culture. And I thought I'd go and look at the business models of how they've done it. And there are lots of different books have written about this, changing the culture of an organization, and they're very similar. The one model is define, align, and manage. And define, begin with an end in mind, teamwork, working together toward a common vision, and then manage, a culture of discipline, a principle of greatness. And I think that Dr. Clayton and the Office of Research in Women's Health is actually going through these steps. Except there's one thing I think that has been left out of the discussion that I think is absolutely critical. And that is, how are we going to measure success? Because if we don't measure success, we will not know if we need to change what we're doing or if we've succeeded. And so how do we measure it? And so the last part of my talk is going to be on the metrics, some suggested metrics, and I think we need to have more conversations about what these metrics really should be. And so for short term, one could easily measure the number of applications and grant awards that are proposing single-sex studies in males and those in females. That's a metric we could measure. What about the number of applications and grants proposing dual-sex studies? We could measure that too. And then we can look at ratios of these. And of the single-sex studies, we could look at the male-to-female ratio. And of the dual-sex, we could look at the ratio of male-to-female animals proposed per application or per grant award. And we can do that because, thanks to the vertebrate animal section that is required on all NIH grants involving animals, you have to describe the animals in terms of the species, strains, ages, and sex and numbers. So this is required. It's not enforced, but it is required, and NIH does say that they can use this as a scoreable point, and it can be withdrawn administratively if you don't address this, and it can affect your score. So we can measure this, and these are short-term metrics. What about midterm metrics? I would say that some midterm metrics that could be a valuable indicator would be the number of published papers on a single-sex study. Again, we can only do what is cited by NIH support, but we could know the number of, we could quantitate the number of papers on a single-sex study that are in male and those in female, and then we could look at the number that are reporting on both sexes, and then look at ratios of dual to single-sex, and of the single-sex studies, the ratio of male to female, much like what this paper we've seen cited earlier today did, where they looked in 2009, the number of animal articles in 2009. Gray is where the sex was not specified. Purple is where they specified both. Red is where they specified female, where it was female, and blue is male only. And you can see there's a lot of blue here. And we could quantitate, we could see whether these numbers improve with all the initiatives that the NIH is going to do. And we could see that this 75% of investigators who don't report the sex of their cells would now, this number would go down. 75% is, there's a lot of room here for improvement. And we can do that because it's not anything new. We have to upload our papers into PubMed if we're funded by NIH right here. To advance science and improve human health, NIH makes the peer-reviewed articles it funds publicly available. We already have to submit our papers online. There's no reason why we couldn't modify this and capture the number of papers that are published that are on males only or females only or in both sexes. And I'd like to think that in the future, what would happen is that this is, again, from that same Berry and Zucker paper. So if you look at the number of articles over time here, and you look at the blue bar, the blue circles are the male only, I'd like to think that if we're successful, this number is going to come down, and the number of single-sex female studies will go up, and the number of dual-sex studies will also go up, and that the number of studies that don't report the sex go down. So that's a measurable metric that we could capture, and then we would know, hey, we're doing what we need to do. And then we could also, of the studies that are looking at both sexes, we could look at what of those papers, how many were actually published a sex difference. So I did a quick survey. I used keywords, sex differences, gender differences, and this is the number of papers published on that year. So 1940, those terms were not used in PubMed, but look at 2010. This number is going up. That's great. That's good. I think that we could expect to see this number go up even higher if we are successful with these initiatives that we are going to introduce. And what about the long-term metrics? Because why are we interested? Why does it matter? Why do we care? Well, I think that the long-term metrics are things like the number of clinical studies and trials whose design explicitly addresses sex-specific hypotheses that have been revealed by preclinical studies, or proposes study and pooled analyses of males and females due to a lack, because we don't have the preclinical sex differences in a study that was proposed to do that. And then the number of established and newly developed drugs with sex-specific labeling by the FDA, like we're starting to see, in the field of sleep with Ambien, and I think we'll see more of those. If we are successful. And then we might see the number of consensus statements and clinical guidelines recommending sex-specific therapeutic strategies for treating disorders and diseases because of sex differences in efficacy and or safety go up. And lastly, reductions in sex-specific skewing of adverse event reporting, which we heard earlier. This is the 10 drugs that many of you know were pulled off the market, with 8 of them having worse adverse consequences in women. And that, hopefully, would go down. So when I was thinking about culture change, I was thinking, what could I point to that would be a great model? And it is the 50th anniversary of our anti-smoking campaign. I thought, this is it. This is the perfect analogy, because we could have stopped smoking in this country if we just said, it's illegal, you can't smoke. But we didn't say that. And that's not what I gather we're prepared to do here, not like we did with the NIH Revitalization Act, and we required women to be included in clinical research. What we're doing here is we're going to motivate investigators, basic scientists, to do sex-based biology and think about sex. So that's kind of like the anti-smoking campaign. It was it was they tried a lot of different things. But I want to make the point, they were measuring the number of cigarettes that were smoked, the number of people that were smoking all along this 50-year campaign. And we have to do the same thing. Because if you look at the kinds of things they did, and they did a lot of things. If you look at this red line, I mean, the first thing they did is they put warning labels on cigarette packs, right? And that was in 1966. In 75, they no longer provided cigarette rations to troops. But look at that, though. The number of cigarettes that are being smoked was still going up. So I'd say that if they stopped there, we wouldn't be where we are now today, which is at 18%, which is great. We have 18% of the people in this country now smoke, and we can get that number down even lower. Compare that to something over 50% when we started out. That's progress, and that's exciting. And that's why I believe that we can do this. We can change the culture. But we have to measure, because if we don't measure, we're not going to know that we need to keep doing things. And you can see all the different things that the anti-smoking campaign, all the things that were tried. And it's kind of it's cumulative. There's no one. I thought it would be preventing people smoking in public places. I thought that would give you the biggest drop, but apparently it's not. It's cumulative. So there's educational initiatives, and there's making it more difficult to smoke. But there's a lot of different things that we've done to try to get people to stop smoking. And I think that what Dr. Clayton and the Office of Research in Women's Health is doing is perfect. They're using educational initiatives, and they're using trying to encourage investigators to, in the grant review process, to address and think about sex. And I think that this putting everything together will hopefully have an effect. But we have to measure it. We have to measure it. Otherwise, we really won't know if we've been successful and if we have to add another component. So with that, I just want to thank Dr. Clayton again for putting this excellent workshop together. And it's been really exciting to me to hear all this great science and policy and thoughts. So thank you.
Thank you, Dr. Sandberg, and thank you, Dr. Miller. I think we have about 9 minutes for questions for our speakers. And hopefully the mic will work. If not, you can come up here and ask. Okay.
I'll meet you halfway here. Thank you so much. So 2 terrific talks and very thought-provoking. And I just wanted to come back with a couple also strategically oriented questions. Catherine, one of the points you made that I don't think we should overlook is that in some circumstances we do not have the right models to go after the questions that we want. Virginia, you also mentioned tools. And I would hope that that would be one of the areas that we would collectively as a group really get our recommendations around, because with the pressure that most scientists are under and most scientific entities are under, having the tools to go forward would be enormous. The second comment I wanted to make, and then I'll toss that back to you, is that what you're telling us is that culture eats strategy for breakfast every day, and we know that. So I think this concept of culture and moving forward, or Virginia, a lot of things that you were talking about, which are cultural also, silos versus networks, et cetera. What would be, in your views, the most important things we might do to be able to change that culture? I think there's going to be a lot of resistance in the community if we mandate that one has to do X versus Y. I could be wrong. But if we approach this culturally, where do you think the best bang for the buck, so to speak, would be?
People change behavior if the incentive is high. So I think a good example of that was in the early 90s when there was RFAs out to look at the effects of estrogen on cardiovascular disease. It drove a lot of investigators into that field. The RFA amount was limited, but what resulted after that on study section were all the unsuccessful applications ended up coming in to different study sections, and there was a plethora of knowledge that came forth from that initiative. So I think, I know money's limited, whatever, but I think strategically placing some of that into some of these initiatives with alternative animal models. We've learned a lot from cellular studies. We've learned a lot from genetically altered mice, from all different kinds of animal models. But I think another place where we need to consider is alternative models for disease other than mice. I mean, we're looking at complex traits. Okay, how much can we really learn more by knocking out single genes in a lot of these animals, or modifying them, or turning them on, or turning them off, when we heard from Art's work that we're really looking at a sexome and clusters of gene and gene networks. So I think we have to have some education in terms of encouraging investigators to look at gene networks. So those would be two areas what I would put forward where there might be helping to change the culture. Catherine, what do you suggest?
Sorry. I was just in a meeting in Brazil, and I was impressed how many investigators there were interested in the influence of biological sex in their studies. And they were all young, and I think if you educate the young people that this is an important area, that you'll probably have the most effective change in culture.
One of the things that I was thinking about, and I meant to mention it in my talk and I didn't, is that I'm starting to view the sex differences as a health disparity. And there's a lot of driving force to reduce disparities in healthcare. And if we consider sex as a health disparity, there is incentive to investigate those differences from a variety. We've expanded the pool of influence in terms of pressures to try to reduce those disparities. And catchwords are fine, but in reality, they are health disparities, and these are a primary target for reduction in terms of healthcare delivery and the purpose of NIH. So I think thinking about these things in health disparity. And we had a conversation with, before we started, about leadership in research centers, and whether the sex bias in leadership at research institutes in terms of not being diverse and having a diverse mix of sexes really biases agendas.
Other questions? So I have a question about how you might see from the outside looking into NIH, because I think that if you look at NIH, it's really hard not to see the silos. And, we at NIH talk about this and struggle with it. But from the outside looking in, how would you envision us to be able to move forward to sort of break down some of those silos? Do you have any real pearls of wisdom where you think NIHers could, really make a difference?
I think if we have initiatives that cross institutes, so that you have, requests for applications in different areas that are crossing institutes, like the NHLBI and NIDDK, for example, then I think that can help break down these barriers where there's cost sharing. And so that would be one way of because in one sense, one institute would only be responsible then for half the amount of money, and they would be able to claim that as part of that research as part of their portfolio. So I'm a strong believer in team science, and I think that could be one way, as Virginia has said, is creating these networks is really, really important, especially in this looking at the influence of biological sex and gender.
I think the structure of the study sections also could be better integrated to have an interdisciplinary program. that tends to happen on program project reviews, but not necessarily for R1 reviews or K reviews. And typically you get individuals with siloed kind of interests and so on without really understanding the integration. And to be able to provide a quality review for a proposal that may integrate multiple systems into a sex difference through some other new kind of resource sharing model.
The idea of a team science approach and breaking down the silos is not just going to take the NIH. It's going to take your whole institution buying into that. And if you look at how the academic tenure and promotion structure is created, there's very much an emphasis that a young scientist prove that he or she can individually run a research program. It's no surprise that many of us over here are fairly senior at this point and have embraced the team science aspect, but junior scientists are getting a very different message from their home institutions about it. And maybe that is also where some of the education institutions.
Yeah, actually I've sat on rank and tenure, and they now actually have a category for collaborative science, and they actually value it. So some institutions have heard that message, and hopefully others will follow suit. But it's actually we have to address how much collaborative science we've done in rank when we go up for tenure or promotion.
So I just wanted to discuss a little bit the silo issue at NIH, and it's more of I don't have any actually solutions because I think it's a fundamental, deep-seated problem. The study of sex differences is actually a discipline by itself. There are animal models and procedures and an understanding of hormones and sex chromosomes, and there is that discipline is not actually represented, to my knowledge, in any NIH institute mission statement. So no one institute will embrace that. If you wanted to, for example, have a T32 to train scientists across disciplines like cardiovascular, neuroscience, immunology on how to go about studying sex and gender issues, where would you take your T32? What institute could you go to? Okay.
Idea.
So those are just questions I'm asking. Certainly when one applies for a research grant on sex and gender influences on a disease, the study section are great experts on the disease, but they're really naive about the sex and gender issues, and it's very difficult and yet, for example, Catherine's study is where the sex of the T cells that you're infusing into the animal, that discovery is interesting not only in the study of hypertension, but it's interesting across disciplines because there's an immune component of so many diseases. And so knowledge of that sex bias in T cells is of great interest, and yet it's not well represented. I'm sure you already know this, and I'm just saying what's already obvious. But it is I think a deeply seated issue in the organization of the NIH. The kind of quote unquote funny way I say it sometimes is no NIH institute wants to cure somebody else's disease, right? Especially in the reduction of funding of research, each institute is focused on a very specific set of diseases. And these kind of broad issues such as sex are kind of lost in the shuffle.
We do have another question back here. I think that might be the last one we can take for the day, but these are excellent questions and responses as well.
This isn't a question. Actually, it's a response to Art's question, but I think it's a really important point, which is as people begin to generate these new ideas and these breaking down of the silos and networks and looking at multiple body systems, there's going to need to be a review process that will be able to meet that problem. And 100 years ago, I was a scientific review administrator actually at CSR. And one of the values of CSR is that it's not aligned. So it has study sections, and the study sections have cultures, if I'm still correct about that. But it's not aligned. And so if there were a way to, for this initiative to also include ensuring that there was expertise in sex and gender on the multiple study sections, for example, and also to ensure a way of reviewing multidisciplinary applications, which is always a challenge. I mean, when I was at CSR, they called them complex applications. And there was a lot of talk about how you review complex applications. But, I mean, there is a mechanism that exists, which is to bring on other experts for those applications. So I think some kind of explicit conversation about this with suggestions to have expertise in sex and gender represented on all the study sections might actually be helpful.
Excellent point, and I see Dr. Clayton is taking all kinds of notes, too. So I think that wraps it up for now, and we have a break, is that right? And for how many minutes? 10 minutes, no more.
Good afternoon, everyone. We're almost finished, but we're not quite done yet. So if you could please take your seats, I'd like to reconvene the meeting. We'll get on with our last few discussion points. This will be the last song and dance of the meeting, so get prepared, get your cameras out. This will be a YouTube event. Oh, Corley, yes, please. So for this session, what we're going to do is this is really your opportunity, and all the people in the audience, all the people in our video audience, and anyone who's listening, this is your opportunity to weigh in. All right? You have heard a tremendous amount of information today. I've learned several things myself, and I'm afraid to admit I've done this research in the past. And I should know this. But I learn something every day, and every day I work at the office, I learn something new with regard to sex differences in women's health. So we have at the front of the room here the moderators from the sessions today. My name's Susan Meyer. I am the Deputy Director of the Office of Research on Women's Health. I come to this methods and techniques issue because I'm a methodologist. I'm sorry. I admit it. But I'm not embarrassed about it because methodology is where it's at, okay? Knowing how to do something, knowing how to structure your experiment, your study, your clinical study, knowing how to analyze the data, what's the appropriate type of thing to do, and how to interpret your data. Those are all things that are very, very important to any field of study. Okay, I think we can all agree on that. We can agree that methodology, techniques, statistics, experimental design, those are all very, very important. We've heard about some things that are lacking. Maybe there aren't models that are appropriate for some of the things we want to do. Maybe males and females aren't necessary. Maybe they are necessary for very specific types of things that individuals are looking at. So this is your opportunity to weigh in. We do have a bit of a structured discussion. And what I'd like to do is I have two, let's see if I can get them. Okay, I can't get to them. We had two comments come in the mailbox. And one comment was a comment on the doubling of the N. All right, and this comment came in from a viewer in Israel. And this person suggested that at some point, we can't just double N. All right, and I'm paraphrasing from them. You can't just double the N, all right? It might not be possible to do so. Scarce resources, you have, you didn't do it that way in the beginning. So what you can do is certainly try your experiment. You can use it with, if you had an N of 8 in each cell or each group, you might try 4 males and 4 females. And then if you don't detect a difference, you're not powered to do so, but you may get a glimmer of something, then you can move on and say, look, I really need to power this to do so. Then you use the same experimental design, the same methods, and you can power it up so that you can detect whether sex has an influence or whether there is a difference or not. So I think this was a very thoughtful comment from this person, and I think we've heard that a few other times today, is that, sometimes understanding if there's a sex difference may lead to more research on the sex difference, or it may tell us something about an underlying mechanism, about something, a method, about a pathology that might not even involve sex differences anymore, but it certainly led to new discovery. And NIH is all about discovery. Okay? So those were I think most of the comments that came in. What was the other one? Sorry, my Outlook wasn't working. Is that it? Okay. Oh, okay, yes. So here are some more questions that came in, and these will certainly be very good for our discussion. So the question would be then, should sex specifications be required for cellular and molecular studies? And one step further, should the field require that both sexes be explored in proof of concept studies of mechanism? And how do we bridge the gap between preclinical and sex effects in humans? Okay, so those were the questions that came in that also came in with the other one that I just stated. So we're going to use these as fodder, if you will. I'm going to have the moderators weigh in as they want to. I'm not going to drive this whole song and dance, but I do have a few things that I'll jump in with. I'd like to go through and do a little exercise with everyone. I think it'll start the discussion, and then I would like to go to the actual call to action, and I want you to tell us how we can do this, okay? Because you're going to suggest what we need to do, how we need to do it, and what's our implementation plan, all right? So I have a question for everyone. All right, answer this question. You have to start your answer with this. So sex matters. We've heard everyone talk about this. So by using both sexes in preclinical research, ... Go. What do we gain? What do we lose? What is it that's a problem? What's the barrier? What's a yes, please?
Okay, I'll take the first leap here. I was struck listening to all the panelists that we've talked a great deal at NIH about personalized medicine. I think I've said to several people in my institute, NCI, we call it precision medicine. We can never be the same as everyone else. However, when I think about the difference between whatever we learn in the research setting and actually observing physicians who are taking care of us, that to me is the model of personalized medicine. Maybe I've been lucky, but it seems to me that very many physicians take into account the holistic approach to what a patient is who's sitting in his or her office. So what I was hearing here today has to do with, I think, the major opportunity, which is to somehow do the research in a manner that really enables, in a clinical setting, physicians to have at their fingertips the kinds of information that does enable them to provide personalized medicine that is at the state of the art. And I'm not sure exactly how we convey, or how readily or how well we convey what we're learning in basic science to the clinical world. Certainly in the cancer side, we have a very difficult time. It takes a very long time to change the practice. So I, having listened all day, really wonder how we could, using this kind of discussion about sex differences as a springboard to improve how we convey what we're learning about the basic science and the differences in the sexes to the clinical side so that we really can realize this goal of personalized medicine. It isn't a matter of sequencing someone's genome. It's really linking the biology. And this, I wonder sometimes at NIH if we do a really good job of that.
Okay, so first of all, thank you for your comment, but we know that you're from NCI because you didn't follow the rules. Okay.
We never do. My director certainly doesn't.
I know. So let me just rephrase it then. So by using both sexes in preclinical research, we need to be able to develop information at the basic science preclinical level that will eventually transform into information that we could disseminate to clinicians. That's the ultimate big goal. Okay, because we can't serve the patient population, which is more than likely half female, by only studying one sex or 25% of one sex in the experimental realm. Okay. Yes, please.
I'll just finish that sentence. I think that you originally gave us sex matters in preclinical research, and then it was to fill in the blank. And I think I'll just summarize some of the stuff I've heard here today. Sure. Try to do it in a nutshell, and that is to say up front that it's a matter of doing good science. And the reason it's a matter of doing good science is because if there's differences between males and females in preclinical research, and they're not detected, then we're gonna draw conclusion errors. And there could be a finding that is true only for males or only for females, so that's a sex-specific finding. And if it's generalized to, if it exists and it's not studied so as to be detected, well, then generalization is made incorrectly to the other sex, and so. And this is true for human research as well. It's not just in the preclinical realm. So I guess I can just speak more generally about in the human realm as well. That and of course at NIH, all human research has to include females, but gender analysis of data is not always done, and if there's an underlying difference and it's not detected, there will be conclusion errors. And so, as I was saying, that effects may occur only in males or only in females, and then they're incorrectly generalized to the other gender. And then there may be cases in which and we heard some today in which effects are opposite in males and females, and if the data are not analyzed so as to detect those opposite effects, then they can just simply wash each other out, and then again we have incorrect conclusion errors. So there were effects in both males and females, but if they wash each other out and the data were not analyzed to detect it, then a conclusion would be that there was no effect. And so, rampant conclusion errors is a big problem if we do not include both males and females and do the appropriate analysis. So that is not good science, and it costs us. It costs us in terms of the research that unfolds, that cascades from studies like that has incorrect conclusion errors. So that's a quick answer.
No, that's great because it encapsulates the using both sexes in preclinical research helps generate better science through appropriate interpretation, better science through a generalizability of the results, and better science through an understanding of the basic biology of males and females, and then the difference between them when the sex-specific or sex analyses to compare the two are performed.
And of course, adding to that, we often hear about the concern about perhaps added financial cost that changes might create. But we also have to keep in mind that does have to be balanced or considered in the context of the cost of doing bad science.
So when does the cost make the most difference? What is the difference between the cost of not including males and females in your experiments or your studies at the beginning, but then finding out later in your translation or in your clinical work that you need to or should have, versus the cost of now having to include males and females in your early preclinical studies and then deciding at some point there is no effect in one, or it doesn't make a difference, or the difference is not relevant to the disease or condition under study. So when should the cost be borne, and what is the relative cost? I think for some of the drugs that have come out of, that have been, I use Ambien as an example, we know what the cost was in the end, okay? So that's a consideration. Yes, please.
I don't think basic scientists necessarily see themselves as part of the pipeline in its entirety. And so I think the chemoattractant that we need is cash. It's capital to start with. And I'm convinced once people start doing the experiments and find discovery, basic scientists and translational scientists will get hooked on the euphoria of new discovery. So I think at the outset, and I really love the analogy that Catherine had to the fight for tobacco reduction and all the little incremental things that happened. I would advocate for, I think that costs are not going to be too rampant, in fact, but I think that's the perception. And so I think one thing that could be done is that all grants that include both sexes and an analysis of that sex would be increased by one module. What that does is it puts a chemoattractant in the water, and people will now all of a sudden want to do this because they think that that will actually fund some of their research in a more provocative way. I also think RFAs are a good way across all the ICs that each IC gets on board and has an RFA that is from each institute. I think the notion of inter-institute funding, as we had for the roadmap, was wonderful. I happen to have one of those common fund grants. It was fantastic, but it's just cumbersome. So I think we need to have each IC, and in fact one of my notes is educate NIH Institute directors. They need to be the champions for the best, most reproducible science, and encourage notes from the IC directors to their grantees about their enthusiasm for this award, together with a link to an RFA from each of them. So those costs then become a we need to neutralize that as part of the argument right now, because as long as the big hand is up, no, I can't do it because of costs, even though the analysis as Londa presented today is ongoing and the costs are unlikely to be enormous, we still have to combat a perception. And so I think those two tactics, three tactics, increase modular budget by one, put an RFA out from every individual institute, and get our institute leadership on board with this, I think could go a long way towards getting us towards that first couple of steps along the pathway that we saw for smoking cessation. I would neutralize the cost discussion up front.
You realize I can't write a check, right? Well, I gladly take your suggestions for it. Yes, please, Lee.
So I think sex differences matter in preclinical research because they really can teach us something about mechanisms that one would otherwise not be able to really ferret out. And that then becomes instructive for doing the clinical research. And as a recovering academic who did clinical work myself for years, and I didn't do very much of the basic mechanistic work. I think I have a ground to stand on, just as having a man in the room speak up for wanting to do sex differences. So I think I can speak for the clinical side of it and say how important it is to do this basic work examining sex differences. And I also want to say something about RFAs in this day and age at NIH, where we have such stringent and stretched budgets. So what I would say is that, we shouldn't let the perfect be the enemy of the good, in that if funding opportunity announcements come out and they're PARs or PAs, that's a good thing too. Yes, yeah. It's okay. It doesn't have to be a set aside. Right. So.
Okay, some questions from the audience, please. Go ahead, Dr. Miller.
So you asked us a while ago what we would have to suggest to NIH to sort of restructure or encourage this. I would like, you're in there, okay? Do you have insight as to what you could do within the structure of NIH or restructure it to improve this interdisciplinary translational sex differences approach? I mean, you want us to fit it into your paradigm. Maybe you all need to fit into our paradigm.
So I guess I just throw out a teaser, and I don't know if this is being recorded, but I would probably, if I was, queen of NIH for a couple of years, I'd probably want to restructure NIH entirely because I think it's very disease-focused, and I would like for us to be much more health-focused. And one of the ways that I've thought we might do that is by actually organizing a new institute. Some institutes would have to go away, probably, but organize it according to what some of us call in the health field as a three-legged stool. So sleep, physical activity, and nutrition. And I think that sex differences work into that paradigm very, very well. But I also think there are other areas that work into that very, very well. And it really becomes so difficult to study a number of health-related outcomes in our current structure because we do, even though we try to reach out to other ICs, institutes and centers, we still live in silos. So I would want to restructure entirely.
So in the absence of restructuring, could I riff on that question? I'm not intramural. I'm on your side, but I think we also have this notion of us versus them, and I do think that as we're putting these notions in the water, we have to appreciate the intramural science and extramural science all have to be singing the same song, although it seems to me that intramural science can be converted now. And perhaps there's some structural reasons why not, but again, I'm just trying to figure out what the soft underbelly of this issue might be. And if Collins can immediately say to intramural scientists, we're working with the extramural grantees because there is a grant relationship that is a contractual one that has to be set up associated with study sections. And so I have a lot of recommendations on that front that I hope we can get to. But right now we could turn the switch on intramural science and say to all the members working in this building, neuroscientists, most of them, you're now going to include sex as one of the biological variables. Now, you're not going to have additional funding because appropriations has already occurred, but begin to think about how to integrate sex and report to us the null data, the same kind of thing that we're talking about in the extramural environment. It seems to me can happen today with leadership from the top. And I think that would go a long way towards starting to think about what are some of the issues that come up in a pragmatic way once this starts authorized. So I do hope this is recorded because I think that would be a really smart thing to turn the switch on. We're not doing it for October this year, which we thought we were, but having a very, I think, good and authentic discussion with extramural grantees to try and convert hearts and minds. Intramural, I think that's ready to go. And that may not be restructuring NIH, but that uses NIH in a very positive way to move the agenda forward quickly. Good comment. Thank you.
So I wanted to go back to this issue of it costing more money, which it may well cost more money, but I was also quite taken with Londa's point that there's a lot of infrastructure already in place, that it may not cost that much more money. And also if you measure it against the money lost by drugs developed that have to be taken off the market and that sort of thing. So I'm wondering if one of our metrics might not be actually to track the money and see if it really does cost more money. I mean, if we, for example, compared, I don't know, maybe this isn't accurate exactly because there's probably studying slightly different things, but let's say we compared Larry Cahill's program on emotion to Adam Anderson's program on emotion. Would one cost more than the other? And one looks at sex differences and one doesn't. So I think that would be a really interesting project. And also maybe part of a metric that would be useful.
But I think there are certainly ways that we could run numbers. And I think anyone could do that. Anyone who has extramural research, intramural researchers, and even us lowly extramural administrators can certainly run numbers on this. And this was as a PI myself with rats and mice and sheep and goats and cats, you always were being cost conscious. How do I make sure that I maximize what I have by not putting in, not spending all of my money just on that one resource? And that's where pooling data, pooling resources, I believe Dr. Sarabji has benefited from that in past years because my pregnant rats that I used for my studies to generate pups, when they become 12 months old, they're now reproductively senescent, okay? So what we do is I say, okay, I'm going to transfer my rats to Dr. Sarabji. Dr. Sarabji is going to work on reproductive senescence and different mechanisms related to that. So pooling, sharing, and being collaborative certainly helps with that. But yes, it will not be difficult to run numbers on that. I think as we start to explore, today a point was brought up about alternative animal models. As we start to explore those different animal models, fish is one of my favorite, but other ones, Drosophila and flatworm and things like this, that, there may be an opportunity to reduce costs entirely. But
yeah, and I just wanted to speak to very much agreeing with Jillian on that, is that we don't need to compare apples and apples as far as programs. You can model that very readily. It would cost you very little to do it. The other thing that wouldn't cost a whole lot, I think, would be really to put out, we've talked about education, we've talked about culture, but this is an issue of what are the best practices in this area. You've heard all day long about how people do their science, how they use their animals, how they construct their cells, how they put their programs together. And those best practices for people who've been working in this field for a long time could be extremely helpful for people that want to get on board with this or feel they should or whatever. And that's something from the NIH that wouldn't cost a whole lot of money. Much less than a program announcement, but highly effective, I would guess.
And I think that's a great idea. And I think one of the questions I had for our actual call to action recommendations was, how do we get, and it's related, is how do we get people to learn about this? How do they know? How do they know what to do? We all know what to do pretty well. I think many of us are experts in the field. But how do we teach others? How do we get that information across? NIH can do different types of activities to promote and to put out information. But I think all of you have the same responsibility to do so, whether you have graduate students, whether you have medical students, and whether you have newer faculty in your departments, that you do also have that particular resource. So developing content and making sure that it is something that can be disseminated by everyone, not just by NIH, but by everyone. It's not our dictum. It's not you will learn this. This is a collective understanding of how we do work, and this is how you might try to do it. But no, great idea. Yes, ma'am.
Well, I'm going to take a contrary view. Surprising, because I think we're sitting in a room amongst, we're in the choir, right? And
yes.
This is obviously an extremely collaborative group where there's a rather strong sex ratio in the room. But out there in the wilds, there's a whole lot of people who are really angry and upset about this, and they're going to do everything they can to avoid it, and they're going to throw up the cost card constantly. And I know all of us, I have heard it from our colleagues, were they going to give me twice the amount of money and this kind of thing. I just want to return to what Teresa said. I think that she's absolutely spot on. We've got to put the money issue to rest immediately. And I think it can be done with a relatively small amount of money, but it can't be a lot of hand-waving of, oh, this isn't going to cost you anything. Because we can bring those horses to water, but we can't make them drink. We have to make them want to drink. There has to be some kind of incentive that says that this is a good thing for me to do. A single module, that is not a lot of money, but it is more than enough to make them drink. And then we train them how to do it. But the one thing that I wanted to ask, though, is, and I know Teresa's ready to answer this, is if we do make it something that is actually paid for, then it has to be a review criteria. Right. And so how are we going to deal with that?
Yeah, I think it should be a review criteria. And I do think that many people in the room are on the Trans NIH committee, Janine, that are working on this. So I think this is part of your call to action.
Yes.
And it seems to me that there's a number of things. Is this my cue to start reading my list? So
please.
I do think that we have to require researchers to report sex of subjects and outcomes by sex and include both sexes in their study. And this is for intramural and extramural scientists. And I've already talked about turning the switch on the intramural now, convert them now, because that can be done. And I do think that we're talking about sex at this point, and I think that curve for smoking cessation is one of the things that eventually gender, or as we begin to define what environmental impact or gender will be defined as in vertebrate animals, that can be described. And I say here, vertebrate animals and cell line sex must be reported. Test sex differences in one part of your experiment, and then if no difference, include male and females in future design to be sex inclusive, and adequately justify this and report this. I'll come down to some tactics for that. Create model systems as a shared resource. I think that's an RFA that could come out. I mentioned the cost. I think we have to train researchers on experimental design that includes sex variables and require that as part of the reproducibility modules that are already being built. So I don't think we have to build differently than that, and that's going to come in, I think, as part of the responsible conduct of training. And so I think sex comes in right in line with that. I think we have to encourage the report of null data. Journals are disincentivized to do this because many of them are run by societies and organizations that need to make money from every single page. And so this needs to come into NIH. And I think there needs to be some thought on how NIH is going to do that, because that needs to be front and center. I think we have to educate the NIH Institute directors, and I've mentioned that they should be our champions. We have to create some RFAs that develop animal models that phenocopy human disease. Again, this is a way of putting money in the water, chemoattractant, for people who may not be within the enclosed circles, who have already done this, who might have some ideas. And I think all those RFAs that was mentioned earlier create grants, and those grants eventually come back in the system. And so every RFA, it's like the $1 equals $140 in biomedical advances. I think you always get a 100-fold impact on RFAs. I think we have to create a sex outcome website with experimental evidence of sex differences, sex equality, and good experimental design, and essentially create a template based on field-specific leaders that have vetted scientific results. And I think we've seen some of those excellent examples here, and we know them, and we've heard each other's talks, but I think we have to put that into a format that then can be used didactically, and I think that can be accomplished fairly readily and should be done with some alacrity. I think we have to educate proposal reviewers and SROs on sex inclusion design elements required, and I suggest that there is one study section member who is assigned to review the sex inclusion elements for the next year, so that there can be some examples of how this should be done. It's similar to when women first came onto study sections. There was one each on each of the study sections after the NIH was sued, just to see how it would work. And I think that this could be accomplished here. I think we have to monitor inclusion policy compliance, both through grantee reporting, but also through biopsies of published papers. So we can roll up the data at the end of the grant, but I think published papers also have to be evaluated. I think we have to get the vice presidents for research on board at all the grantee institutions. There should be a video meeting required for all VPs that have NIH dollars. And this sounds very punitive, but I think that they should be required to listen to the issues on reproducibility because their institution directly benefits from the NIH dollars, particularly the way we've talked about how tenure is structured, and we're putting it on us, but really institutions have structured salary based on how on NIH budgets. Usually 80% in clinical departments are required from NIH budgets. So I think they can be incentivized by fairly strong language, but we have to do that because then we've got another pain point, and some are going to be chemoattractants, some are going to be pain points, and all of these things are then eventually going to keep us from smoking too much. We have to encourage institutional research center cores to identify services that focus on sex variables. 2 times ago in the Office of Research and Women's Health, there was a discussion on institutional cores. And if cores start having these kinds of activities, that again is a little pressure point that then each individual scientist who is feeling so under the weather over everything that's happened from sequester and, you know, not knowing if people care about them, scientists need ego stroking all the time, you know, at least this is a place that we can go and find information, and we can get information out to those cores, I think, relatively quickly rather than kind of the hand-to-hand combat. The FDA, NSF, pharma, biotech, all the agencies really need to get on board with this, and that has to come from Collins. Journal editors, we've already talked about, and I think, as I mentioned earlier, all NIH press releases must include the sex of the animals in study. So NIH has to start reporting on the variable that they think is important. And if there is no sex reported, the NIH doesn't do a press release, even if it's a big deal. So that starts to incentivize NIH to actually think that this is important. Medical school and graduate school curriculum, professional societies are the ones that develop the clinical guidelines that then the physicians use. So many people in the room help with those guidelines that then go to the patients. And we're encouraging through our public advocacy for patients to come in and say, was that drug tested in a sex-specific way? You know, and that's a mouthful, so we need a pithier way of saying that. But that's a really important way. Even though that physician is sitting down and talking with you one-on-one for your 10 minutes, it doesn't mean that they know whether or not the sex-specific guidelines have been followed, and I think that's a real gap. So the consumers increase awareness on the importance of sex and medical research to consumers, but not in a way that's punishing. Is everybody tired of this? We can't conflate sex inclusion with cost, and we've talked about that. And journals need to accept sex inclusion data as authentic and not boutique. Everything can't go to the Women's Health Journal, even though we love it. But that's just not the right way for science to continue. And so those of us who are on editorial boards, or, you know, we've really got to get on board with figuring out how to get these things there as authentic science, which it absolutely is. My final thing to say is that our end goal, I loved all the metrics, is that our end goal is that we should eliminate the Office of Research in Women's Health, because we have done such a good job of inculcating all of these kinds of metrics for women, for minorities, so that these kind of health antecedents that are developed through basic science pipeline are all so there, Janine, that we don't need to be there. That's our goal. Our end goal is to have a party to eliminate ourselves. So those would be my recommendations as I've sat listening to this today.
Thank you. Thank you very much. Does anyone have any additional things to add to Dr. Woodruff's list? Okay.
I just had a comment.
Yes, please.
I agree with everything you said. The use of the word chemoattractant, you know, the extra money. I keep coming back to that issue because
That's
it sounds
pejorative.
to me,
It's too pejorative. I apologize for that.
And
yes, sorry.
It also suggests that this is the sugar coating on a bitter pill. And we have to get away from the idea that this is a bitter pill.
No, it can also be a sex pheromone, so you can come.
However you'd like to phrase it.
If we go back to the,
right.
if we go.
I think the messaging has to be very, very positive. And
it is positive.
That you will
so
learn
yes.
If you include it. And that here is, of course, the availability of funds is going to be important. I agree with Julian's idea that we need to actually see what the increase will actually cost. I mean, some of this may just be in people's heads because they envision doing everything over twice with 4 pairs of hands now to do all of this, whereas in reality, you can very strategically incorporate it. So I think the messaging needs to be very positive, and it needs to also not mainly speak about how women were disadvantaged by this, but also how both sexes are disadvantaged by it.
Right, an important point, yes.
I just wanted to make one comment that is sort of an additional thing that program directors could do at NIH. You know, if we are in the process of developing funding opportunity announcements that are not, you know, specifically on sex differences. So just funding opportunity announcements in general. I think we need to be including in those FOAs the language and being able to really frame the issue so that in each one of these FOAs that come out, that we really want to examine sex differences. And so I'm trying to think of a way I can do this for an upcoming series of funding opportunity announcements. And I think I might, talk to Janine and figure out if I could even have her come and talk to some of the other program officers as well.
Yes, sir. Can you get
I may have missed it, but
yes.
I may have missed it, but in the various recommendations and suggestions here, did anybody talk about having program officers review their portfolios to see if the portfolio as a whole was improving on this dimension or some metric of inclusion of both sexes?
I don't recall the program, actually portfolio analysis itself,
Yeah.
but certainly a review of what it is that's available, what's out there, what have we done, and how does it, interestingly, how does it vary by institute and center?
Well, yeah, that sounds a little different than like having each program officer be measuring in some way their portfolio with regard to this topic. And I think there's at least some program officers out there who are going to be quite interested in doing this if they are released to do this by their superior officers.
Try to address that a little bit, because we just, at NCAM, we just went through almost a year's worth of portfolio analyses, whereby each one of our program directors had at least one portfolio analysis to conduct, and that was dictated by, and I don't mean that in a negative sense, but it really, we were told what type of analysis was wanted from above. And I think that if what you're talking about could work, I think it would have to come from above, because, and I cannot tell you the amount of time these analyses took. They came out of our hide. We weren't released from doing anything else in our job description. So it was weekends and evenings, and it's a lot of hand picking through the journal articles. It's enormously time consuming. So I'm not saying we couldn't or shouldn't do it, but I think it has to be thought of really in a very logical way from above and with a lot of discussion. But it's a good point.
I echo that on behalf of my colleagues in the NCI. We were asked to do the same thing. And just as you said, we have the progress reports rarely have any information like that, except in the cases in which someone is studying on the basic side an animal model or a phenomenon that we know has been shown in the clinic to be a sex difference. And the other was having to go through publications. Now, we weren't asked to look at our entire portfolio. That, at least in my division of the Institute, with 2,600 grants, that's a lot of work to do. But I can also tell you that we are looking at, on this committee that we have now, at how we will be asking researchers, applicants in the future, to define how they are going to do their research with sex differences or sex in mind in the research design. We are talking about ways in which we might be able to automate the process of scanning the reports so that, in fact, we could report on an annual basis how many grants had, in fact, adopted a sex type of experiment design so that we have both sexes. But that is not a trivial thing to do. But one of the things they said was, I think across the institutes, most program directors would quit, because if you had to do that every single grant that comes in, we have enough trouble getting compliance with putting the publications into PubMed.
But could you not just ask that be part of the progress report, require that it be part of the progress report?
that we can in the future.
Right, and not in the past,
But
retrospectively,
going forward. Well, not just retrospectively. It takes us a couple of years to change anything in these forms because of the Office of Management and Budget. But but many of those things were started a couple of years ago. Trust me.
So to just echo that concern about the laboriousness of getting that information if it's done by hand. So the problem is the lack of an automated system, whatever that system might be, and of course that whatever form it would take would be the ideal solution so that this could be tracked. And unfortunately, it's non-existent right now, but maybe moving forward we can think about doing things differently.
Are there other questions or issues you would like to bring up before I start on my list? No? Okay, well, this is the song and dance part, okay? So I'm going to give you, in a nutshell, basically what we talked about today, okay? Just to give you a refresher, try to pull you back to where we are. I know it's late, but bear with me for 5 minutes. So I started out by asking, by using both sexes in preclinical research, what is it? What's the benefit? Okay, I think we heard that you may be able to decrease variability. You can account for variability associated with using both males and females. You have the potential to see an interaction of sex with your variable of interest. You can't do that if you don't include both sexes and if you don't design your experiment well. You have the opportunity to decrease sample size. We know that may not be possible for every single condition or disease, but you have the opportunity to do so. And as the literature builds up and you know what your effect size is, you have the opportunity to start to reduce your sample size. So you have the opportunity now also to interpret your outcome, and it's meaningful. So what does your outcome mean if you just have treatment and control, if you don't know if it's male, female, young, old, something, something, red, blue, black, or green? You have an opportunity to increase power by your sample size. If you're using 5 males right now as your N in each cell, and you add 5 females, then you've got an N of 10 in each cell instead of an N of 5. How do you do that? How do you control that variability? Well, you can control that variability by knowing females have an estrous cycle. I can monitor that estrous cycle. I can know how many females are in which portion of that estrous cycle. I can use my 2x2 design to be able to account for the variability from the estrous cycle and build that into my statistical analysis. I can increase generalizability by including both males and females in my studies. I might only want to use one sex. There may be a time when there is only one sex to use. Why would you do this? Well, we know that studying both sexes helps understand the biology of both sexes, okay? We know that there could be a difference. Males and females are different on something. Studying one sex can certainly help us understand that sex. Okay, that sounds trivial, but if we think about drug development, if we think about diseases that are very fast-moving, contagious, if we know that one sex is involved and we know that we have to develop something quickly, using only that one sex is a time that's justified, and it allows you to cut down on the time through to drug development, discovery, and hopefully treatments for sex-specific conditions. So that's a sex-specific type of analysis. We can only study sex differences or the differences between males and females when we collect data on males and females. So this is part of leveraging. So we're leveraging resources. We talked about, it costs so much. It might not cost so much. We're not sure what it costs. But if I don't have females and males in my initial study, and then I decide that I want to do another study, I need to be able to know whether there's an effect on the males and females from the original study. I can't do a study all in females and then a year from now do it all in males. That you're going to have issues with confound and with different cohorts. How do you compare those cohorts? What if a male experimenter tested your mice on in the first experiment, now female, test your female mice in a second experiment, okay? It's all rolling up into experimental design and thinking about the questions you're going to ask. You might only use one sex in the case of very, very scarce resources, engineered animals, primates, large animals, animals that are very difficult to generate. Anything that's embryo lethal, you can't generate those, or it's very difficult to get the outcome that you need to get sufficient sample size. We have the opportunity by studying both sexes in preclinical research to understand the mechanisms of pathology, physiology, and function at a very, very early stage. So by using cell cultures, by using in vitro work, by using explants, by using all types of tissue culture techniques in which we know the sex that the tissue came from, we have that opportunity to build that in. We can move faster by understanding that there is a sex difference, there is not a sex difference, or it's important to look at both sexes when I move from my cell culture to my animal work and on. The outcomes in sex-segregated cultures can lead to precision. Medicine can lead to precision. We lead to better identification and appropriate development of our targets, appropriate view of what is it that's happening out there and what should I look for, because my cell cultures are very specific. Cell cultures are not the ideal, they're not perfect, they have their own issues, but combined with the knowledge, combined with knowing the limitations of what your model system is, whether it's in vitro, in vivo, or other, you answer that question by doing your studies, doing your experiments. We can actually use both sexes to understand each sex. So we can vary the hormonal cycles. We understand what females what cycles females have. We can control those cycles. We can also manipulate those cycles to understand questions of relevance. So ultimately, using both males and females in our preclinical research leads to better science. I think we all agree on that, or there's no disagreement I'm hearing from the audience. The interpretation of our outcome when we include both sexes is clear. There is a difference, or I'm not sure yet, but I'm going to pursue this. Using both sexes in our studies leads to better science, because even if we don't find a sex difference, or we find a sex similarity, there's no difference, we might discover something else. And that's all about science, is how we move forward in a discovery loop, a discovery process. Leads to better science through the use of multiple creative and innovative experimental designs, repeated measures within subjects' designs. We can reduce the number of creatures that we use by employing these designs and then applying the appropriate statistics to determine whether there is a difference, whether there isn't a difference, or whether there's a similarity, or we can answer a question at a reduced effort, reduced cost perhaps. And we end up with an understanding of these mechanisms, as I mentioned earlier, if we start to study them earlier. So we don't study whether females and males differ in phase 3 clinical trials. We want to step back and take it back to the very, very early studies, even starting with cell cultures. So I think all of this, as everyone has articulated in this room and those in the audience, in the video audience, everyone believes that sex matters. And I think, for lack of a better tagline, and since I'm running out of time, hang on, sorry, we can do this. We can do this, and we can tell others how to do it, and we can help them through whatever means we do, whether we issue money, whether we provide extra funds, incentive, whether we show people how to do it, whether we develop educational materials. We can do this. Yes, ma'am, please.
Susan, that was terrific. That was an outstanding summary, and there are just two things, since it's being recorded for utility down the line, that I want to just state. The first thing you mentioned that we talked about estrus cycles, but we didn't talk about them in terms of having to do them, as you suggested. And I would urge Janine to get a podcast with Irv Zucker or Brian Prendergast so that they can describe to the scientific community about these pesky hormones. Second only to cost in people's minds about why they shouldn't do this is the cycle. And that paper definitively helps us all understand that in general biology, we can study males and females independent of the cycle. Once you find a difference, you're going to want to go down that path. But I wanted to clarify that, Susan, in your comments, and I would urge the office to get Irv or Brian to do this ASAP. The second thing that you mentioned, as was mentioned here, was the earlier today, the issue of the individuals who are handling the animals as part of this gendered part of the equation. And I also think that's going to be third on the list of why people are going to be concerned about this issue. And I want to point out that I suspect that if Jeffrey Mogul were here, he would also say that was one study with one endpoint that had to be about pain. That has not been replicated in another lab, and it has not been replicated in any other biology. And so the biology of pain receptors, we know, is touch sensitive. And, you know, so there's all this pheromonal things that he is uncovering in that paper, which was hard fought for to even get published. So I think we don't want to generalize about the issues of management of the animals in our report as you listed. It is something to be aware of, but it is not something that I think we want to have as a generalizable part of the dialogue at this point in time. So those were two things that I'd like to just correct in the motion.
Absolutely. Thank you very much. Does anyone else have any other comments before I turn this over to Dr. Clayton to close the meeting? Last time. It's the song and dance time. No? Okay, that's it. Dr. Clayton, please.
This has been an absolutely fantastic day. I am really so grateful to each and every one of you for your participation, in particular the folks up here. But I have to begin my remarks by acknowledging Dr. Meyer and Dr. Miller, who worked incredibly hard along with Team ORWH to achieve this meeting without support from contractors. So please join me in giving them a round of applause. Dr. Meyer said everything that I was going to say in much better than I could say it. So I'm going to say two things, okay? And she's doing a dance back there, so that's a good sign. I'm going to talk about two things which are near and dear to me as a physician and are why I'm at ORWH rather than at the National Eye Institute where I started out. First, do no harm is the tenet of physicians, and I want to talk about first things first and beginning with the end in mind, okay? So beginning with the end in mind, that just makes sense, right? I think somebody had it on their slide. Did you have it on your slide, Virginia? Okay, Catherine. Okay, first things first and beginning with the end in mind. What are we trying to achieve? Okay, so you refer to the NIH mission. So often the first slide of all of our talks is the NIH mission. What do we do here? Well, I usually say the H in NIH is for health, unlike some other agencies. The mission of NIH, as you all know, is to enhance understanding of basic living systems and apply that knowledge to improve human health, minimize suffering, minimize disability, and enhance quality of life. If that's what we are doing, that's our goal. When we begin, we need to design our experiments, we need to select our research question with that in mind. Human health. Humans come in two basic types. That's at the most basic level. When you're born, the first thing they say is it's a girl or it's a boy. That's the first thing. So you got to start with first things with that in mind. And if we are to do that, we think about it, you consider it when you're developing your research question. If you're developing a research question on a disease that affects men and women, you need to consider that there might be similarities or there might be differences. You need to use the power
of a comparative approach to get the most out of every single experiment that you do. Every dollar is so precious right now. We have to do as much as we can with every single experiment. And we could do more if we thought about this in advance, if we took on a different mindset. That's the other word I want to leave with you. Culture change we definitely need. I have to say, when I talk to scientists, when I start talking about culture change, I think they zone out, just to be honest. We need a change in mindset. Scientists are thinkers. That's what we are at our core. We look. We try to understand. We look again. If we have a mindful, purposeful beginning, why am I doing this? What is my research question? Let me design my experiment to test that. We could learn so much more, because that's why we are here at NIH. So I left ophthalmology to do this because I thought there was a chance to do something more than a very circumscribed, highly specialized subspecialty in ophthalmology. And this is so exciting to me because we can make a very big change. And while she stabbed me in the heart a little bit there, Dr. Woodruff saying, we want ORWH to go. I'm just teasing. Of course, the ultimate success, if I were queen of NIH for a day, Dr. Alakel, ultimate success would be that everyone does this automatically. Then I could do ophthalmology. I don't know. So, of course, it would be that we're not needed because this is part of science. And I agree. The next generation, the early stage investigators, they should be the change agents. They are amazing. You guys work with them all the time. They're incredible. They should be the change agents. We do need champions in our leadership, absolutely. And I'd be happy to continue meeting with them and talk about this. But I hope that I can count on every single one of you to be with us in this challenge and in this fight. I think we still have a fight. I think we need to be very thoughtful and strategic about how we move forward. And I think we need to understand how the game is played, how life works, the realities of what the pressures are on people in academia, the realities of what NIH can do, the realities and opportunities of partnering. We cannot do this. None of us can do this alone. We need new partnerships. And you're talking about a multidisciplinary team versus an interdisciplinary team versus a real team versus combining some ingredients and putting together, thinking you're making a cake. We know the reality. We know what collaboration is. We know what sharing is. We know what cooperation is. We know what getting together just to put something together and call yourself a team is. That's not what we need. A team means everyone is interdependent and you're working together for a common goal. And we can do this. I agree with Dr. Meyer. I'm going to end with her comment. We can do this together. Thank you very, very much for being here. Thanks also the communications staff who've been tweeting and doing interviews in the back, Anne Rancourt and Bonnie Tabasco, and all of the speakers and moderators. We are so appreciative of all your time and effort. Thanks very much.